Hello, check. Hello. Papa, Papa, dago. Hello, check.
এই শঙ্কর, hey, I'm surprised to see you.
Oh, I'm surprised to see you. Where are you? Are you in Texas or in Calcutta?
Texas.
Okay.
শঙ্করদা,
হ্যাঁ,
সুব্রত, P47 KDA protein functions.
আমার
মনে পড়া
মাথাটা এখন পুরো জ্যাম হয়ে গেছে, অ্যাকচুয়ালি গোবরে ভর্তি হয়ে গেছে।
ফিলাডেলফিয়ায় ছিলাম, P47 KDA প্রোটিনের ফাংশনের কাজ করতাম, আপনার সাথে কথা হয়েছিল তখন। বড় সুব্রত আমাকে এই নিয়ে একটুখানি কথা বলতে হবে তোমার সাথে।
কতদিন আগে এটা কথা বলছো?
আমি তখন এটা অলমোস্ট 27 ইয়ার্স ago আমি তখন ফিলাডেলফিয়া ছিলাম, অনেকদিন আগেই ছিলাম, এখনো কিছুদিন আগেও গেছিলাম। এখন বোস ইনস্টিটিউটে শেষ করেছি, রিটায়ার্ড, এমিরিটাস সায়েন্টিস্ট, ইন্ডিয়ান কাউন্সিল অফ মেডিকেল রিসার্চ।
এখন কোথায় তুমি কোথায় থাকো কোথায়? আচ্ছা আইসিএমআর।
আইসিএমআর এমিরেটাস সায়েন্টিস্ট। হ্যাঁ
আচ্ছা তুমি কোথায় থাকো? কলকাতাতে থাকো?
হ্যাঁ কলকাতা থাকি আমি। আমার বড় ছেলে এখন নাকি সল্টলেক সিটি ইউটাতে আছে, ইউনিভার্সিটি ইউটা এট সল্টলেক সিটি।
আচ্ছা আচ্ছা
ওইখানে আমি গেছিলাম, কদিন আগে গেছিলাম, আমি ফিরেছি, বেশ কিছুদিন ছিলাম।
আচ্ছা
But it's not
সল্টলেক
yet.
সিটি। যাই হোক তুমি তুমি ইউ আর রিলেটেড টু স্বপ্না?
রিলেটেড টু স্বপ্না, আমি রিলেটেড টু অদিতি দেবনাথ। স্বপ্না আমার থেকে অনেক জুনিয়র।
Oh, I see. Oh,
আচ্ছা
I see.
অদিতি। কজন এটা how many people are showing up at this meeting?
I mean, and the lecture probably six speakers are there probably.
Yeah, I don't know. শঙ্কর, এই শঙ্কর,
শঙ্কর দাকে ডাকছে।
তুমি কবে ফিরছো কবে? I don't think you can hear me. You can recognize Shankar audio?
From pictures.
Yes.
Yeah, I'm sorry, I was muted.
No, you are coming back. We are returning to the.
No, I'm leaving tonight. I mean, this evening.
আচ্ছা তুমি কোন ফ্লাইটে এসেছো? কোথা থেকে আসছো?
No, Doha, Qatar. Qatar Airlines.
This is the Qatar
Yeah.
airline. Qatar, yes. How was the trip?
Trip was very crowded. Otherwise, it's fine.
তোমার শরীরে কিছু প্রবলেম হয়নি তো?
No, I'm doing fine. Yeah.
Great.
Yeah.
পাপাকে
How is
বলছি,
the weather there?
হ্যাঁ ওয়েদার এখানে এক্সট্রিম কোল্ড ফ্রন্ট ইজ কামিং।
Oh, is that right? I see. Oh, but
That's Kendrick.
Oh, hi, you have become bigger, taller. Yeah, I see. Hi.
Much taller than me, of course, but that's nothing saying much. That's not saying much.
Okay, this is my nephew,
Oh, acha, by Kendrick. This is
my computer assistant.
সম্পাদ ছেলে।
Some,
ওর নাম কি?
My nephew, my sister's son,
ও তোমার নেফিউ? না তোমার এই সম্পাদ।
your brother's son, not
Hello.
হাই
Baipo.
ভাইপো, আচ্ছা। না তুমি এখনো কলকাতা বাড়ি ফিরে যাবে, তারপরে তোমার ফ্লাইট রাত্রিবেলায়?
রাত্রিবেলা না, আমি একটা হোটেল, this is a hotel, I'll go back from here after the talk.
আচ্ছা
But these two end at 4 o'clock, right? I
আমার
can't
টক শোনার জন্য এলে?
Huh?
না তুমি তোমার টকের জন্য কটার সময়?
আমার talk তোমার পরেই।
পরেই? তাহলে তো ভালোই
Yeah,
হয়, তারপরে
Right, and I cannot stay till the end of the. Yeah, it's okay.
আমার তো মাঝরাত্রি, তুমি তো
তোমার অনেক টাইম রাত্রি তো।
রাত্রি 11টা অলমোস্ট।
Oh my goodness.
So my talk will be the end of
If
my
you want to hear the whole talk symposium, then it will be 3 or 4 o'clock.
participation, huh?
If you stay till the end of the symposium, it would be 3 or 4 o'clock.
Absolutely. No, there is no way I can do that. I'm way too old. I was going to say my opening statement is I'm the oldest person here in this audience or anywhere close by. But you have somebody came close. That's where you.
What is that? What are these people? Is it in Maliganj Science College?
I have no idea. Oh, তুমি বাড়িতে আসছো? Shankar is also from home or from a hotel in Calcutta.
Yeah, right.
তুমি কলকাতা হোটেলে তো কোথায় আছো? এই
Park Hotel.
কলকাতা পার্ক হোটেল।
Park Hotel. Park Street.
Park Street Hotel, Park. Now, I had, Bimal Bachawa, Bimal
হচ্ছে না তো?
Very few. Yeah.
Maybe you and I are only two of us.
Yeah, I think so. I think there was the guy who used to work with Dave Baltimore, but he retired from science. What's his name?
Who was there, Baltimore?
No, that guy who was there in that meeting, he retired.
Oh, yeah.
Inder Burma.
I've heard of Dave Bolton.
Yes, he got a Nobel Prize for reverse transcriptase. Yeah.
Right.
I've read
right.
in that one book.
Kendrick wants to be a biochemist.
Oh my goodness. There is no job available nowadays. But it's a good idea. You'll do well.
I don't know. Well, you're not interested in medicine? Doctor, don't you make a lot of
Are
money?
you
They're
more
not interested.
medical type or basic research type? He wants to do.
He can hear you. Speak a little louder.
I wanted to enter research as well.
Okay, good. Yeah. Clinical research.
and then starve.
And then starve. That's the
Oh,
next one.
wow. I see. Okay. Wonderful. Okay.
আচ্ছা আশিস ফোন করেছিল আমাকে
What is sharing?
Screen sharing
Yeah, let me- Okay, I'll try to put on my slides as a trial. He'll help me.
Okay, go ahead. No. Take your time. Do that first.
WhatsApp audio.
Hi. No, that's okay.
Or actually, yes, look at that. He's also Shankaradia. I am S, there is an S, there is an S, there is an S.
Everybody knows.
Exactly. She is Aditi Nag Chaudhary.
Not in S.
No, that's not a no S. No S there.
Not a simple one.
আমার এটা অফ করে দেয়নি তো আমার অফ করে দেওয়া উচিত
No, they can hear you're talking about S.
হ্যালো এভরিবডি করে দেবেন তো অন করাই থাক হ্যালো এভরিবডি প্লিজ টেক ইউর সিটস গুড মর্নিং টু এভরিবডি প্লিজ টেক ইউর সিটস উই আর গোয়িং টু স্টার্ট আওয়ার প্রোগ্রাম Hello, everybody take your seats and please switch off
Hello.
your mobile. Please, hello, all the guests present here, please switch off or make your mobile to silent
Aditi?
mode.
Can you hear me?
Your
Yeah,
mobile should
or
be in
like
silent
this.
mode
No.
or
Aditi.
switched off. Please.
Can you hear me?
We are going to start. We are going to start.
It's frozen.
Good morning to
Or
everybody.
maybe no one is.
Huh?
I don't know. Everyone else is muted. So.
Everyone else
Professor
is muted.
Mitro. Professor Mitro, can you hear me? Hello, Professor Mitro.
What happened? Everything is frozen,
Hello, can you
No.
hear me?
but nobody talking.
Huh?
Well,
It should,
oh,
huh?
Yes. Can you hear me
never
now?
mind.
Hello, Dr. Mitro, can you hear me now?
Never mind.
Hey, Shankar, what is happening?
Okay. Sir,
I'll
I
be
am
back in
inviting you
2
to
minutes.
deliver your inaugural speech.
Okay.
Then Professor N.C. Mondol will introduce you, and then
Hey,
we'll deliver
Aditi?
your special lecture for
hear
today's
me.
program. Am I clear? Am I clear, sir?
Yes, microphones are turned on or turned off.
It's on. Everything is on. Everything is on.
That indicates
Everybody
muted.
can see you.
Yeah, she's muted.
Everybody
But
can
she's
hear
not
you.
muted.
You start your inaugural speech. Then Professor N.C. Mondol will introduce you, and then you will
Hey,
deliver
Taposh.
your special lecture.
না আমি ওদের কথা শুনতে পাচ্ছি না ওরা কি বলছে আমি তো জানি না শঙ্কর বলছে শঙ্কর ক্যান গো বিফোর মি দ্যাট উইল বি ফাইন
Hello. Deliver your inaugural speech.
ও শি ইজ টার্নড অফ and yeah Aditi cannot hear me.
Hello.
None of them can hear me. Can hear anybody.
Can you hear me?
They are both no. Aditi is not turned off.
Yes. My mic is turned on.
I'll mute you for now.
My mic is turned on.
Taposh.
Yes. Yes. You can
Taposh is on the phone.
No, you hung up.
start your inaugural speech. Please.
It's already 10 o'clock.
Who is B? BC Grow Hall.
That's what
Please
the meeting
start.
will be.
I cannot hear anybody.
Can you hear me?
Yeah, I can hear you. Hey, Taposh. না আমি কিছু শুনতে পাচ্ছি না আমি দেখতেও পাচ্ছি না আমি অদিতিকে আমি দেখতে পাচ্ছি but I cannot hear her. Now the phone I don't know. Anyway,
Can you see me now?
Just be patient.
Hello, Dr. Mitru, can you see me now?
there's a snafu. Gauri is in the audience.
I see.
Shankar, you're not in the audience. I'm not in the audience.
I'm just gonna start camera. So I think this is like an audience, a spectator
Okay.
account.
Okay. So I can start.
Sure.
Please do.
But who?
Somebody will introduce you. Wait, just be patient.
হ্যাঁ না বলছি আমাকে কেউ ইন্ট্রোডিউস করবে ইন্ট্রোডিউস করা কি আছে আমি জানি না তোমার সাথে গৌরী কথা আছে গৌরী অডিয়েন্সে রয়েছে
এই তো অদিতি
স্টার্ট করতে বলছো
Start.
ওকে অডিয়েন্স দিস ইজ অন ওকে অডিয়েন্স ইজ ক্যান হিয়ার
Start your
Anyway,
inaugural lecture.
it's a great
then
pleasure
NCM
to
will
give
introduce
this talk
you, then your
to honor
special lecture.
Professor Jagat Jiban Ghosh. We were the second batch student of the Biochemistry department in Calcutta University Science College, and Dr. Ghosh or Jagudda was the first hired chief professor in the department. So I give a little background. I don't know how many of you know or know at all, but I just thought I should give a little bit of background. Next slide.
We cannot see this slide. There's no slide.
Oh, you cannot see this slide.
No, it's okay.
Okay. Next slide. Okay. So before I start, I should give an introduction of my guiding principle or whatever you call it. One is serendipity and the other one is introspection. Introspection is, I think, people judge themselves whether they're doing things right and wrong. But serendipity is something which is beyond your control. And many times it happens to all of us that serendipity, you make a decision about a guide, about the future, and you never realize whether this is a good decision or bad, or this is good, helpful for you or not. That happens only after it is done, it's over. And serendipity works a great deal of decision-making help in my life, and that's the reason I wanted to make that point. So I give a little background. First of all, I need to thank Aditi and the Centenary Committee, Jagat Ghosh Centenary Committee, for inviting me to this Reminiscence about Professor Jagat Ghosh. And now a historical context that I, along with 7 others, we are the second batch of biochemistry students. The first batch graduated in 1958, and we are the second batch, got a master's degree in 1959 after 2 years of master's program. And I have written a lot of stuff just for my own benefit, but you cannot read that thing and you don't have to read all of it. But I will leave these slides with Aditi, so you can, if anybody wants to anybody's interested, you can take a look at this later. So with this Department of Science, Biochemistry was in the department, within the Department of Applied Chemistry, and that's in the Baligan Rajabajar Science College. And that's where it started. So I was, historical context is that nobody expected me to join the biochemistry department in the second batch, because biochemistry was just a beginning department and nobody had any idea what the department will be like and what quality of students and what quality of teaching will that be involved. So the hierarchy was that I should have been doing chemistry department because of my academic ranking, which was I ranked 5th in the school final, 7th in the intermediate, and I topped the class of BSc of chemistry honors, and so they didn't expect me to join the biochemistry department. But this is what the serendipity, I made a decision that I should come to biochemistry, and actually I was the trailblazer in the sense that after me, Shankaradu, who has become far more famous than I did, as ever was, he joined the biochemistry department basically following my footsteps. Next slide, hang on. Next slide.
That is the next slide.
Okay. So in hindsight, my study in biochemistry was prophetic. In 1950s, the age of molecular biology has not even formally started. The central dogma of molecular biology, the information transfer from DNA to RNA to protein, was not formally presented and proven until a few years later, and in the late 60s, in the early 60s, and the goal of biochemistry was limited to clinical chemistry and nutrition. And that's what we was taught in the biochemistry department, or the biochemistry division of the applied chemistry department. It was actually way far behind the humongous growth and enthusiasm in science of biochemistry. Professor Guho had a training in chemistry and nutrition at the Cambridge University, and he recognized the full potential of the biochemical studies of diseases. And so he expanded the biochemistry as a distinct discipline. Biochemistry was until then was the Department of Applied Chemistry, and again, as I said, is a very conventional clinical chemistry type. The faculty came from applied chemistry, and the first lecturer appointed in the biochemistry department was Professor Jagat Ghosh. Jagat was the first person appointed, although there is originally the Department of Applied Chemistry has 2 other professors, Sushil Bose and S.C. Roy. But there are other teachers like Professor N.C. Ghosh, I.B. Chatterjee, and Gaurachand Chatterjee. They all came from outside the department, but eventually became a faculty in the biochemistry department. So in my career, I started, it can be, again, it's a question of serendipity, and in distinct phases of my career, I did my undergraduate and graduate studies in RNA, from 60 to 63, from the undergraduate studies, before that undergraduate in India, but I did the characterization of viral RNA, and from 64, 65, I did a postdoc at Stanford for DNA synthesis in vitro. 66 to 71, I did a radiation-induced chemical and physical alterations as by the studies of, again, touching on molecular biology. And then 71 to 92, I studied DNA repair and mutagenesis in alkylated genome damage. And then 92 to 2013, I studied oxidative genome damage, and that's how I ended at my career. But initially I had a stroke of luck, or whatever you can call it. I did 2 papers. The first paper in my life was in Nature, as a Nature note, and that was the paper we worked with Professor G.C. Chatterjee, and that is a paper in enzymology, lecithin as in Vibrio altor. And the second, another Nature paper also from my Calcutta University, from Bose Institute in Calcutta, also in bacterial metabolism and membrane permeability of M13 phage, which is again not very exciting to me now, but that was at least what is successful in helping my career. Now coming back to the memories of J.J. Ghosh, because I worked with G.C. Chatterjee and my Vibrio cholera project, and so I never ended up working for J.J. Ghosh, because I was also coming to the United States, joined the Madison University of Wisconsin for graduate studies, and so I never worked with Jagat, and was busy setting up. Then I went back to India after my completing my PhD and postdoc, 1966, and then I had to start my lab in Bose Institute, and that's why I was too busy and I didn't get a chance to spend enough time with Jagat. However, we interacted socially. It turned out that Jagat's wife and my wife were classmates in undergraduate college. So in 1971, I came back to US and learned about Jagat's professional activities from his students. And we stayed in touch, and we, particularly Shankaradi, who actually will be talking after me, often sent him popular science articles for his use in classroom lectures. Going back to the golden age of molecular biology, we started my MSc course, and after the structure of DNA was solved, then the molecular mechanism of information transfer was solved, and postulation of central dogma of molecular biology. Professor Ghosh, the first official lecturer of the Biochemistry department, taught nucleic acids in the class, and I also took the class coming on molecular biology and nucleic acids. And he stimulated our interest in molecular biology, definitely contributed. My interaction with Jagat included visiting his home. He was unmarried and got married after I left India for my PhD studies. Jagat taught us the course of molecular biology. I didn't have him as a research guide, but on the other hand, he was a faculty guide and supervisor, actually, during our tour to South India. I don't know that they still have too many students, but we are just, because there are 8 of us, our batch students. So we had a very excellent tour of South India, Belur, Halebidu, and so on. And Jagat was a delightful company, particularly for barely adult boys and girls. We had 4 of us, or boys and 4 girls. 2 of them did not show up for the tour, so there are 2 girls and 4 boys went on the tour with Jagat. Jagat is about 12 years older than I, and often talked to me at least, as an equal, particularly during those long train rides. He has an excellent sense of humor. We all laughed at his jocular comments, reminiscing about the snippets of his comments. I recognized afterwards that he was born to be a teacher. Jagat was born to be a teacher, an excellent communicator who could effectively paraphrase complex concepts in layman's language. After studying biochemistry, including nutrition as a student at Calcutta University, I switched my interest to newly emerging molecular biology, and Jagat's lecture in my chemistry class helped illuminate the challenges of this nascent field. Professor Ghosh's unique strength I'm sorry that I'm switching between back and forth between Jagat and Professor Ghosh. Professor Ghosh's unique strength as a teacher was in highlighting the key concepts or phenomena, and thus help in conceptual clarity. Back to my own research enterprise, although I started studying molecular biology of RNA for my PhD thesis, I became a maven in DNA structure and function after spending 2 years at Stanford and carving out my own niche in genome damage and subsequent repair to restrict the original sequence of the genome. Genome damage and repair, the relevance to diverse pathologies is being recognized more and more. Almost in every disease, there is a component of genome damage and altered cellular signaling or cell death. And that kind of thing has actually was a major pathways which still are being worked out, after because of the variety of genome damaging agents, genotoxic agents, and exogenous methylgenic and genotoxic agents, as well as the endogenous genotoxic agents. And the most common agent is reactive oxygen species or ROS, and they are generated endogenously as well as exogenously, and that I spent rest of my life actually studying the signaling and genome damage caused by reactive oxygen species. So I joined. Is somebody going to say something? Hello? Anyway, I joined after I went back came back to the United States. I said went back. 5 years in India. I spent 5 years, and I produced several students who got the PhD, and I left India, and I found a job at Oak Ridge National Laboratory, which is founded by Alexander Hollander. And this is a radiation research lab, and I was interested in radiation. And the people who are responsible for my joining there are two of their top scientists, Audrey Stevens and her husband, Sulil Neogi. Sulil Neogi was my good friend from Calcutta University. He was a applied chemistry, biochemistry student in applied chemistry. And because of him and we moved back to US and with a job at Oak Ridge National Laboratory. And then from there I moved to University of Texas Medical School in Galveston. And Watson was responsible for Sam Wilson, who unfortunately died recently. And Sam recruited me and a few other people in the DNA repair studies. And he was absolutely dedicated in DNA damage and repair and the repair enzymes. He was a super biochemist, and he did a terrific job of building up the department. Unfortunately, he left and went back to NIH, and the department actually, and many of us left, so that suffered badly in this future of the department. I will not talk about the science because I just will mention few discoveries that we made in our lab and key persons and the collaborators. So we studied with the methylated bases and genome damage, and we cloned the genes using a phenotypic complementation or interspecies complementation. And I was lucky that I had a lot of people in my lab who were very dedicated and motivated. And starting with initially in Oak Ridge, like these people, I cannot point out, can they? They can see my cursor? Okay. Anyway, so the people here are the people at the Oak Ridge National Laboratory. The two came from Japan, Ikeda and Shioda, and Tano. Three came from Japan. And then others are local. And the Nepean nucleus with Izumi and Bhakat, and you can see the names. So these are the people, various people who were collaborated. And I'm very proud of the fact the most important thing I did in my life was to nurture and groom the fresh PhDs and people motivated and stimulate them or inspired them. They could do great science themselves on their own. I'll come back to that. The people who are successful scientists who came out of my lab. How many minutes have I spent?
We have like 15 minutes.
Okay, 15, okay. I'll finish in half an hour total, I think. So we also studied other, the previous page showed the major activities, but it also did a lot of other studies, ancillary studies or relevant studies, and including covalent modification of BR proteins. Many of these are discovered in our labs. And again, these are the active people, the junior scientists, physics, postdoc, mostly postdoc. Some graduate students also did a lot of work. And he also had a collaboration with my colleagues, John Taylor, and used to be in Lawrence Berkeley lab, or used to be in anyway, at Lawrence Berkeley lab, and now he's at MD Anderson next door in Houston. And you can read what I'm going to say, but so I decided that you can let you read this. So there is a lot of stuff is still to be discovered. And there are many other challenges, and that's one of the problems people will face. And these are the people who joined our Silly Center for Molecular Science, which is founded by Sam Wilson. As I said, Sam died a couple of years ago. And then Stephen Lloyd moved to Oregon State, and Ben Van Houten moved to University of Pittsburgh. And Ben was a graduate student at Oak Ridge when I was one of his thesis committee members. And John Taylor also recruited Albino Bacola, who is a bioinformatics guy, and did a great job in our studies. He collaborated with John Taylor, as well as without John Taylor, with Albino Bacola. And then I'll mention some of the people who work in either my collaboration or my guidance, and then I try to help them develop career as an independent scientist. The best example is István Boldog. He's a Hungarian. He worked in a lab as a glorified postdoc, and Sam Wilson asked me to talk to him and see whether he can help him, and we did, and he got a career, he got an award, a grant, NIH grant, and he became independent. And I did some politicking. I don't mind doing that if it's a good cause. And he has been a very successful scientist. Steve, nickname is Steve. And we are still people are collaborating with him. They published a lot of papers, and you can read this thing, all these things that he did accomplish. And then Tadahide Izumi is a Japanese postdoc. I spent 3 months in winter time in Kyoto University Radiation Institute, and I recruited Izumi to finish his PhD there. Kyoto is one of the top universities in Japan, and then he came to my lab. Izumi now he's migrated and settled in the US, but he did a lot of work, published a lot of. He's a very smart guy. Unfortunately, he's having some problem with grants, and just like many other people in the US. And so I'm hoping that he'll be successful, and I have a lot of faith in him and a lot of expectation from him also. He's a great scientist. And then Tapas Hazra, some of you may know him already. Tapas is here in the audience, not in the audience. Tapas is watching it from his home. He just called me. And he has been a very successful scientist because he discovered a whole new class of enzymes called NEAL, which you name NEAL, and they're tremendously important for a replicating cell, replication-linked genome damage repair. And he did a great job, and he's still doing that, but he's also extending his studies into genome damage and repeat sequences and so on in neurodegenerative diseases. And then Robin Roy, also 1 year ahead of Tapas, he did a great job also in alkylation damage studies, and he now a professor at Georgetown University, the Cancer Center. And Kishore Hazra, I'm sorry, Kishore Pakat. I forgot. Sorry, Kishore, I hope you are not listening. And Kishore is also very independent and there's a lot of interesting things, and he's also doing pretty well. And then I also had a Polish guy from Bartosz Steczny from Wrocław in Poland, and he worked in my lab for 5 years, and he did published his specialty in mitochondrial genome damage and repair, and then he moved on to other studies, signaling by hydrogen sulfide and so on, and he's settled in Galveston University of Texas Medical Center. He's still center from, no, I think he's in pharmacology department, some department in Galveston. And finally, it is Muralidhar Hegde, my youngest and maybe perhaps arguably the most successful scientist came out of my lab. He published a lot of papers with me, and he published a lot of papers on his own, and he has done a terrific job in analyzing and merging biochemistry. He has a very solid biochemist, but at the same time molecular biologist and then cellular biologist and whole animal studies. He built up a lab which is really great. He did a great job doing that, and I'm sure I expect a lot more from him in the future. So my take-home messages are absolutely necessary for genome damage repair is absolutely necessary, because DNA is continuously damaged by a plethora of genotoxic agents, particularly those produced by ROS. But besides ROS, there are other agents also, many metals and so on do that. And in fact, ROS is produced during chromatin unfolding by tetoxidase and other enzymes, demethylases and deaminases. Multiple mechanisms to restore the genomic integrity and overlapping repair processes. And the other, maybe the reason is overlapping processes is because cannot rely on only one pathway to repair, because that fails, that the cells will die. So that's maybe the reason why multiple processes and makes it life very difficult for us, for scientists to unravel the whole process pathways. So finally, my acknowledgement. I forgot why did I use a red color for highlighting several things, but now I cannot. My problem of some ability. So these are the people who came into my lab. Something is happening in the background. Okay, and then the left is my collaborators. I had a whole bunch of collaborators from all over the world, and I traveled throughout the world, not because of collaboration, but because I provided the opportunity to collaborate with a number of people. A few people I should point out, like Yoko Akao at Emory University. He was the one who guided us to the NIL2 enzymes, and then there were several others also. I still collaborate with Alan Tompkinson in Albuquerque, and Barn Kaina, I had collaborated with a bit, but then again, I don't. Of course, I quit science, retired 5 years ago, so it's not important anymore. But we still remain very good friends. And finally, to thank people. The person I need to thank who saved me to make a presentation and make the complete ass of myself is Kendrick Tunnis. That's his picture. Much smarter than I, and responsible for completing this presentation. He happens to be my grandson. And this 17-year-old also aspires to be a biochemist. Thank you very much for listening. Now I'll stop here.
Let me start করব it is my yet
if you ask a question, please speak right. slowly because I cannot hear it properly.
It is my great privilege to introduce Dr. Shankar Mitra,
I just want to know, make one statement
an alumni of this department.
that I do not know how many of you, I mean,
He-
when we started, that was almost 60 years, 70 years ago, right? Almost 70 years ago. So I do not know how many of you know the history of biochemistry department, the genesis, and what happened. We missed the middle part because I was not here. And of course, I left India permanently in 1971. But people who are here should at least have a history and should have a complete historical context of these happenings that are of profound significance of the department.
হয়েছে? বলি শেষ করে দাও।
নিতাইয়ের কথা শোনা যাচ্ছে না, ক্যান্ট হিয়ার নিতাই।
Could you repeat that, Pranam?
নিতাইয়ের কথা শোনা যাচ্ছে না, তোমার কথা শোনা যাচ্ছে।
আমার কথা শোনা যাচ্ছে, নিতাইয়ের কথা, নিতাই? You can speak loudly. Nitai.
Dr.
There's no connection.
Dr. Mitra had a brilliant
Oh.
academic.
Anyway,
He
there is
passed
still time because Shankar will be
BSc Honours in
Shankar Raju is
Chemistry from
speaking now,
Vidyasagar College,
and he
standing
can
first
give you
in the first class.
fill up.
Again, in MSc, he stood
Yeah,
first,
into the-
first class first. He then worked during 59-60 with Dr. G.C. Chatterjee on Vibrio cholera eltor and isolated
Not yet.
the enzyme lecithin splitting enzyme. And it was reported in Nature at that time. He
That's
received
Nitai,
his
yes.
PhD degree in 1964 from University of Wisconsin, working on viral RNA characterization in the laboratory of Paul Kesberg.
So I'll close now
Eh?
and Aditi can introduce the next speaker, Professor Addo.
Okay. Then he did postdoctoral research on in vitro DNA synthesis at Stanford Medical School in the laboratory of
Speaker is
say
there,
it
but microphone
Arthur
is not
Goldberg,
working.
who is a Nobel laureate during 64-65. He came back to India in
Aditi, please,
1966.
Samra, and you please check the microphone
It's supposed to be
at the auditorium.
Excuse me, but nobody can.
Can't hear Nitai.
It's okay.
হ্যালো 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 স্যার শুনতে পাচ্ছেন? হ্যালো হ্যালো
Oh yeah, you cannot see anything. Yeah, this is a problem, Snag. I have had bad experience like this in not too many times, but few times.
Can you hear Nitai? Shankar?
No.
No. We can't hear
He
anybody
did his
from the auditorium.
postdoctoral research on in vitro DNA synthesis
That
at
is why I
Stanford
suggested
Medical
that you
School
should
in the
make sure
laboratory
that
of
everything works.
Arthur
in
Conway,
84
I
or
say,
earlier stuff.
who is a Nobel laureate. Then he came back
Turn it off.
to India and joined Bose Institute, the Department of Microbiology, and stayed there till 1971. He worked there on radiation-induced
Not
damage
yet.
on DNA using
এই মুখটা দেখা
single-stranded
যাচ্ছে তো
DNA
বলতে
bacteriophage
চেষ্টা করছে।
M13.
Aditi.
He then, in 1971, you know now, he went back and joined Oak Ridge National Laboratory.
I cannot hear anything.
Work.
You can,
I
you
will
can, you
not.
can
I
start.
will not repeat this
Shankar,
work. He
earlier.
done
No, no, but
remarkable work on
I think that is
DNA
something.
Good idea,
damage,
because they have some
DNA
problem
repair
with the,
and damage.
auditorium microphone.
Isolated the O6
If
methyl
he's talking, he does
guanyl
not know
methyl
that.
transferase
That I can
enzyme and characterize. He also cloned the human counterpart of this gene and studied it. Anyway,
The other guy is on mute.
Huh?
There are lots of people helping him, so that's that.
Let me see.
Dr. Mitros group also,
Aditi,
Dr.
can you hear
Mitros
me?
group
Raise your hand
discovered
if you can hear me.
that
Can you
the
hear me?
enhancement of
Aditi's microphone is turned off.
chemo or radiation sensitivity of
Yes,
tumor
microphone is turned on.
cells using targeted DNA repair inhibitors. After the early studies on MGMT, that is methyl transferase inhibition,
Surprised that he
he
keeps on
continued
talking without
research
speaking to the
aiming at
speakers.
identified inhibitors for repair pathways for oxidative or radiation damage in DNA. He has around 120
এই শঙ্কর তোমার ফ্লাইট কটায় রাত্রিবেলা
120
আজই
So
publications
it's okay. It's
and
plenty of time.
many
Yeah,
book
okay.
reviews and book chapters and has
নিতাই কথা
collaboration,
বলে যাচ্ছে কিন্তু that nobody is hearing.
good number of collaborators
But why he's talking?
and
Because other people
PD
try to help him,
associates
he does not.
from different corners of the world. He was elected a fellow of American Association for the Advancement of Science in 1989 and a fellow of the Japan Society for Promotion of Science and participated in lecture tours in Japan in 1991, 1999 and 2008. He was awarded Mark Brothers of Indiana Prize in 2009. Dr. Mitra has been continually funded by NIH since 1982 and has been serving on various NIH and other review panels. Now I will tell something with my association with him. I joined Dr. Vishwa's laboratory at Bose Institute in 1965, and Dr. Mitra Shankarda joined microbiology, the sister department next door, in 1966. There is a lot of sharing of instruments with me, and so we are acquainted with him very. Now, we heard about him, but I saw him in my own eyes. I have দুটো ইনসিডেন্টস আমি বলবো। একটা হলো আমাদের যে একটা জার্নাল ক্লাব ছিল, সেখানে ছাত্ররা খুব ভয় পেত, শঙ্করদা কখন কি প্রশ্ন করে বসবেন, এই ভয়টা ছিল আর কি। আর 1967 যখন গিলবার্ট এবং এরা ওই রিপ্রেসার পিউরিফাই করলো, ফার্স্ট এভিডেন্স দ্য রিপ্রেসার ইজ এ প্রোটিন, ওই পেপারের উপর আমি জার্নাল করে বক্তৃতা দিতে চেয়েছিলাম, কিন্তু ল্যামডা সম্বন্ধে আমি তখন ওয়াকিবহাল ছিলাম না। তো ওনার কাছে বললাম যে শঙ্করদা আমাকে ল্যামডাটা একটু বলে দেবেন, লাইফ সাইকেলটা শর্টে। তখন উনি বলেছিলেন, তুমি ল্যামডা সম্বন্ধে কিছুই জানো না, কী করে লেকচার দিবে। আমি তখন বললাম যে শঙ্করদা, আমি তো জানি না বলেই আপনার কাছে এসেছি, আপনি সেইটুকু বলে দেন, তারপর তো লেকচারের দায়িত্বটা আমার। তো লেকচার দেওয়ার পরে উনি, হি ওয়াজ দ্য ফার্স্ট ম্যান, আমাকে এসে বলছে ওয়েল ডান নিতাই, ওয়েল ডান। সো ইট ওয়াজ এ কমপ্লিমেন্ট ফর মি। আর আরেকটা কথা বলছি, যেটা হলো উনি যে 71 চলে গেলেন, আবার উনি আর ফিরলেন না। এখন তারপরে 1974 ডিপার্টমেন্ট অফ বায়োকেমিস্ট্রি আলাদা তৈরি হলো বোস স্ট্রিটে, উইথ দ্য পোস্ট অফ শঙ্করদা ফ্রম মাইক্রোবায়োলজি, পোস্ট অফ সুধামণিদা ফ্রম কেমিস্ট্রি অ্যান্ড পোস্ট অফ বিবি বিশ্বাস ফ্রম বোটানি। এখন সেই সময় আমি ফিরেছি বাইর থেকে, আই ওয়াজ লুকিং ফর সাম জব। তো I got an appointment from Standard Pharmaceuticals as a biochemist. But that time, Shankarda decided to stay there, not to come back. So that post become open now. So I applied and got this post. So I continued my job. That is thanks to Sankoda. Maybe this is for me, it happened. With this, I just thank Sankoda for many times association. পরের দিকে আমার কাজের থ্রু দিয়ে ওনার সঙ্গে অনেক পরিচয় হয়েছে। যখনই এসেছে আমার সঙ্গে ডেকেছে, আলোচনা হয়েছে, প্রণদার বাড়িতে একসঙ্গে ডিনার করেছি, এইসব হয়েছে। ওই দিস আই জাস্ট স্ট্যান্ড আপ। এটা তো পাঠাতে হবে, হ্যাঁ দেখিয়ে দেবো কি করে দেখা দাও কি করে দেখা। PhD with in genetics from the University of Wisconsin, Madison, USA in 1960. Research at the University of Rochester and at the Stanford University, where he worked with Alan Campbell, whose name is associated with jara virology পড়েছে তারা জানবে, with lambda profit integration model by the name Campbell's model, that Campbell. He came back to India in 1969, joined the Bose Institute, where he stayed for 2 years. He then went back to USA in 1971 and joined the developmental genetics section in the Laboratory of Molecular Biology at the National Cancer Institute, NIH, USA, where he now is the chief of this section. Along with the above assignments, he has been an adjunct professor in the Department of Genetics at George Washington University since 1987. Now,
and thanks Aditi and the organizer for giving me a chance to pay tribute to our beloved teacher, Dr. Jagat Jiban Ghosh. So I worked in his lab for my PhD degree, and this brings me some nostalgia. When I was deliberating about the next step of my career after completing my undergraduate in chemistry in Vidyashagar College, I was thinking what to do, and one of my seniors, a previous speaker, Shankar Mitra, suggested that I should get enlisted in the nearest department of the Calcutta University, the Biochemistry department. He particularly talked about as he mentioned in the previous talk, the major attraction there, the new lecturer of the department, Jagat Jiban Ghosh. Since Shankar was already a student of the department, his suggestion was ostensibly honest, and I heeded to it and got enrolled. But do I rule about it? Now the department was far from complete then, and had a newly appointed lecturer, Dr. Ghosh, and part-time head of the department, Piresh Chandra Guho, and about 15 guest lecturers borrowed from other departments and institutions around the town. With the hindsight of more than 60 decades, I say that it was an exciting and rewarding choice. Dr. Ghosh not only effectively coordinated the entire program with skill and dexterity, Obviously an arduous task, but more importantly, lecture in a major part of the MSc curriculum, an inspiring way. Whatever was the modus operandi of his teaching, he was keenly effective. Inside the classroom, he did just not talk, he taught us and made us understand. The way he explained Pasteur effect was as interesting as his presentation. And he also explained Paul Doty's melting of the DNA in a way that made me more appreciative of the double helix structure of Watson and Crick. And that DNA structure influenced me to start studying genetics and molecular biology in 1960. It was a pretty structure, the DNA and the replication mode. The lectures are replete with background stories of scientists and important discoveries. And enamored, I approached him after the final examinations that proposed to join his for a doctorate degree studying ribosomes, and he accepted the proposal. After I had my own lab later, I worked on basic science of gene regulation, as Nitai explained, on and off, how do genes are turned on and off for about more than 50 years. Only during the last 2 years, during the COVID years, I started trying to do something applied, translational research, and which I like to present for the remaining time I have. Now, I tried some nutritional research, which I will describe now, but I don't have the time, so I'll talk only about one of the topic. Now, how do I change the slides? It doesn't change. I thought.
That's okay. What do you want? This one?
Yeah. How is this one? Okay.
No, Jesus Christ.
Just a second. Sorry, I am having problems with the slide. Be patient.
Which one is it? PowerPoint or Microsoft Word?
No.
PowerPoint.
Yeah. Okay.
Okay, you're gonna have to keep going back and
Okay.
forth. That one.
Okay. So, yeah, right. Thank you. Yeah. It's okay. Thanks. So I will talk about our current efforts to make vaccines inexpensively. The word is inexpensively. One slide introduction to Immunology 101. The immune system in our body develops a defense against poisonous substances like bad bacteria and viruses called antigens, which stimulate immune response by producing either protein molecules, specific antibodies, we call them, by B cells, or specific T cells, which become killer cells for the particular antigen. Both trained antibodies and killer T cells are usually made forever in our body and neutralize the poisons when encountered in future. Now, ancient vaccines, sorry, the spelling is ANC. Ancient vaccines kill bacteria, viruses containing the antigens were introduced into our body. That was the system that elicited immune response. But there are dangers, and there are more modern methodology available now, and more modern things are like RNA vaccines and so on. But the problem that the entire process of making vaccines is very laborious, time-consuming, and expensive. We thought of another way to make vaccines to overcome all of these problems, use of bacterial viruses or bacteriophage to deliver the vaccine. Now, my current team is phage-based vaccine. Are the two people, Cynthia and Lee, postdocs shown on the left, and Meredith Schauler, a student, and we are currently at the National Cancer Institute and National Institute of Health, USA. Lifestyle of bacteriophage in bacteria. When a bacteriophage invades a bacteria, specifically bacteria, not mammalian host, it injects into DNA into the intercellular space. This DNA, and normally viruses, bacterial viruses, make a decision and replicate and make more viral components and lyse the holes, and intact progeny phages come out. But there are some viruses which have different lifestyle. They may decide not to do anything except they get the chromosome injected, chromosome is circularized, and sorry.
Need help?
No, it's okay. sorry, and get integrated in the progeny chromosome, and they remain as prophage. When the prophage encounters that the bacterial, because of DNA damaging agents or other killing agents, are in trouble, the prophage come out and replicates again and line the apoptotic growth. But that's not the topic here. This is our choice of bacteriophage to deliver vaccine is bacteriophage lambda. In the picture is the gel structure of the phage is shown. The top is a octahedral head structure inside which DNA is located. There's a tail at the bottom, but there are tail fibers and so on. And when bacteriophage lands on the bacterial surface, the DNA is ejected to the tail. And in detail, the octahedral phage-based bacterial display system is shown here. The icosahedral head has many triangular surfaces, as you know, and in the center is shown one of the triangular surfaces. They're composed of two kinds of proteins, E and D, in multimeric forms, and it is the D protein used to make protein fusions. The sped D protein are fused genetically to protein antigens that you want to make vaccines, and they are fused on the D protein as shown on the right. Now, interesting thing is that the D protein is, I'll explain the later, the D surface protein on the phage head has 455 copies. So it's a copy number. So you have, in one protein phage molecule, you have 450 antigenic molecules to make it multivalent. This is the fusion in cartoon form. The lambda D protein has a aminoid. You can use both amino and carboxyl terminal ends of the D protein are exposed to the surface of the bacteriophage, and we can fuse either the antigen by a glycine-sealing linker, either at the amino end of the D protein or the carboxy end of the D protein as shown by cartoon form here. And the strategy for developing phage display-based vaccine is the follows. First, identify the critical antigens. Sometimes they're available in the literature of the poisonous substance, then display the antigen on the phage surface using the D as a vector protein, as I showed in the previous slide. Then administer the phage displaying antigen into the body of appropriate routes, either internally or intravenous, or depending on the type of disease, and then test for vaccination. That's our strategy. We are trying to make vaccines for several infectious diseases with some collaborations. Two of them are described here. For example, again, the vaccine project goes in the disease. So our idea is to make the vaccine, phage-based vaccine, and inject into the host and provide data, that is how are they raising antibody in the injected animals. Perform mouse study to show that there is an immune response. And also we have the opportunity to make a research, that is we can fuse different parts of the antigen to make it more effective, or either they put the C terminal end is better than the carboxy terminal end. Now, this is a commercial slide. I'm showing why we believe that might not be true, that advantages of phage-based vaccines over traditional methods. First, phage acts as an adjuvant, extremely safe, and they're non-toxic to animals. They don't do any harm to if we inject into the body. There's no side effects. For example, normal vaccines are sometimes made in eggshells, in chicken eggshells, and they are not injectable or not administrable to people who has egg allergy to eggs or something. So the phage doesn't have those kind of problems. Another advantage is simple and very inexpensive. We can produce large-scale amount of vaccine without any real cost. And most importantly, that phage-based vaccines are extremely stable, does not need any refrigeration for shipment and storage. And the last, which I mentioned before, that these are 450 copies of each antigen molecule per phage, one molecule of phage or phage particle, and that gives a multivalency that a very small amount of phage-based vaccines would be effective compared to protein-based vaccines. I'll describe two projects. One of them is a collaboration with Dr. Jonathan Curtis at Brown University, lambda-based vaccine, malaria vaccine. Dr. Curtis, identified a malarian antigen from an African population, I will not go into detail, called PBSEA1, and which is 276 long, and he has shown by protein expression vaccine system that when expressed in, this antigen is expressed in infected red blood cells, and there is specific antibodies to protect against malarian infection. The problem he has is purifying the protein in large amounts and make effective animal experiments. So we helped him by displaying the PBSEA vaccine or antigen into phage and see how the phage works. And this was done in collaboration with him. On the left panel, it shows the lambda displaying the PBSEA vaccine is highly immunogenic. When injected into monkey, these are not our experiments, done them by them. That is on the left shown in gray bar. They have very high titer antibody shown on the left. But if you use instead of display phage, if you use the inject into monkeys, the wild type, which you call normal lambda, doesn't have the display, hardly any vaccination or antibody titer is shown. On the right is shown a real experiment. You take those monkeys and expose them to malaria parasites and look for parasitemia, that is that cause disease. If you, after exposure to pre-exposure of the phage without the display, you see the mosquito infection causes, as shown in triangular thing, causes huge amount of disease, is rising amount. Okay, the percent is about 40% of the infected monkeys get malaria. If you have the lambda vector phage not displaying anything, is pre-infected or vaccinized. Now, whereas if you pre-infected before malaria infection, mosquito infection with the lambda carrying the PBSEA antigen, then you see much fewer, not completely eliminated, 5 to 10% development of the disease. So that result shows that the lambda displaying the malaria antigen is highly immunogenic in animals. And after that, you take over. We have done our job. So we're recently thinking of potential cancer vaccines. Cancer is initiated either normally by infection of an oncogenic virus like papillomavirus, in which case one can raise vaccines against potential antigens of the virus. You identify the papillomavirus antigens, for example, and display the phage. That's one strategy. Or cancer is initiated by a mutation of a critical cellular protein which lets uncontrolled cell or tissue growth, like glioblastoma in brain cancer. Glioblastoma is created by various mutations in the brain chromosome. 30% of the glioblastoma is created by specific deletion mutation of the epidermal growth factor receptor displayed on the brain cells, called EGF version 3. It's not EGFR8, but is a mutant version of called EGFR version C that causes cancer. So this is shown on the right side is shown the normal EGFR. The amino terminal end is at the top, carboxy terminal end at the bottom, and parenthesis on the top shows the, for example, here to there are the surface exposure of EGFR receptor, which binds to epidermal growth factor. On the right is shown the deletion mutant, that part of the I cannot read it, about 270 amino acid, 73 amino acid, and left over is 6 is deleted, the surface exposed part, the internal part is same. Now, on the left is shown the wild type. When EGF receptor binds to the receptor region to EGF growth factor, it normally is inactive. When growth factor binds it controls cellular growth. In the deleted mutant, which causes glioblastoma, the surface exposed part is mostly deleted, and the rest of the intercellular part causes constitutive expression and causes uncontrolled cell growth and which causes cancer. Now, the strategy is to clone this part. That is the part I'm showing. I hope you can see my cursor. That is the mutant sequence which controls only fewer amino acids, and that's we display on the phage. The idea that if we raise vaccines against the mutated portion, it would bind and inactivate those mutant cells, but would not affect wild type cells, assuming that the antibody raised by the mutant version of the exposed EGFR would not interact, those antibodies would not interact with the wild type sequence. Otherwise, it would not be effective drug. So fortunately, EGFR version 3 mutant cells, mostly in brain cancer, 40% of them, has that, and it rarely happens in any other tissue like lung or liver cancer or anything else. So this is the structure with EGFR wild type normal shown on the top part. This part is the exposed part on the cell surface of brain cells, and this part is the functional, which is intercellular. So in the mutant cell, most of the exposed domain are gone, only short part is exposed in the sorry. Yeah, thank you. So the 268 amino acid deletion per EGFR shows the structure on the right. Mutant receptors are unable to bind the ligands, so make it constitutively inactive and has growth advantages for tumors, that causes tumors. And like I said, 20 to 50% of the glioblastoma are caused by this deletion and never found in healthy tissue, which is a good thing for developing therapy.
Ramen or RL Lottie?
Sorry, can you? We displayed the phage on the phage, the deleted extracellular version of the EGFR version 3 protein, not the main functional domain, only the surface exposed deleted part, and injected into mice and tested mice serum for interactions with normal EGFR versus mutant EGFR peptides in ELISA assays. The idea that the antibody raised by injection into this would interact only with the mutant EGFR version 3 protein in the cancer cells and not with the wild type EGFR. These are the results that we are using on the left panel shown. These are ELISA assays. Extracellular, the target is extracellular EGFR version 3, which is deleted mutant version. On the right is the normal wild type extracellular EGFR. And we tested the antibody raised by mice. Okay, I would not go into detail the different versions of the EGFR version 3 cancer cells. On the left, if you injected the buffer into the animal, that buffer, the serum with the injected buffer does not interact much with the EGFR version 3 peptides. The next two, or I'll just describe the Y version. I said that we can use the amino terminal fusion or carboxy terminal fusion, let's say N-terminal. This line, it shows that this interacts, this serum made by injection of extracellular version 3 domain can interact significantly with the extracellular version 3 peptide. And on the right is shown the normal extracellular EGFR does not interact at all with any of the injected serum by injection of any version. Okay, that suggests that the antibody raised in animals by injection of the deleted cancer-causing antigen can interact with the cancer-causing peptides and not with the wild-type peptide, which is a good sign. Which is next slide shows the Western blots. I will not go into detail. The result is that on the rightmost panel, if you had injected buffer or lambda vector without the display antigen, the serum does not interact with the peptides. On the left 4 panel shows that the qualitatively, at least by this assay, interacts with the specific peptides shown in the middle panels of each slide, each panel, and not with the wild type EGFR, which is very clean experiment. So the conclusion is that fusion phage injected serum antibodies bind only to mutant EGFR version and not wild type EGFR, and vector phage injected serum antibodies do not bind either version of the EGFR. And I think that's all the results I have to mention. And let me sorry, that's okay. Thank you. So that's the end of my talk. I just wanted to pay again what a great teacher Dr. Ghosh was, who's influenced many people's career, including me. And that's the end of the talk. And if you have any questions, I'll be delighted to answer. Thank you.
Go back to
Yeah.
the presentation where it's this.
Yeah, thank you.
Oh, I don't think they saw your presentation.
A memento, sir, from the Foundation and Department of Biochemistry, Calcutta University, for you. It will reach to you, and the arrangement will be made by the foundation. A memento for you.
Say it again.
Can't hear you.
Thank you. Thank you, sir.
Can you hear me?
Yes, we can hear you, sir.
Is that black for me?
Yeah, I think so.
Sir, we can hear you.
Can we see? Okay.
I don't think they're checking their messages.
Thank you, sir.
Thanks, Shankar, for this.
Thank you, sir, for your inspiring lecture. Thank you very much. Sir, can you hear us? Okay, sir. Okay, sir. I think you can unmute. Okay, sir. Professor Adho, you can unmute yourself and you can speak with us.
Hello? 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 Hello?
Sir, can you hear me?
Hello? 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 Hello?
Sir, can you hear us?
Here is an announcement. We will come back after the tea. A 10 minutes, maximum 10 minutes break. Maximum 10-minute break. Please come back within 10 minutes for the next session.
না এখানে শুনতে পাচ্ছেন না এখানে মাইক্রোফোন করা আছে ঠিক আছে অন করো এই যে মাইক্রোফোনটা নাও এই যে
না
Hello, check. Hello. Hello. Hello, 1, 2, 3, check. Hello. Hello. Hello, sir. Can you hear us? Hello. Sir, can you hear us?
আর তো নেই অনলাইনে তো আর কিছু নেই প্রোগ্রাম
Hello, check.
একটু ওনাকে ফোন করে বলে দিন হয়ে গেছে লগ আউট করে দেবো না না ওখানে লগ আউট করে দেবো দেখতে পাবেন না তাহলে এখান থেকে বেরিয়ে গিয়ে লাভ নেই এটা রিজয়েন করে দিচ্ছি আর এখানের মাইকটা আমরা একবার চেক করে নিচ্ছি আলো এই বলছি যে তুমি তো গুগল মিটটায় জয়েন আছো আমি এইখানে দাঁড়িয়ে একটু কথা বলছি দেখা তো শুনতে পাও কিনা নাহিদ আছে ওখানে আছে ঠিক আছে আমি নাহিদকে বলছি ঠিক আছে ঠিক আছে নাহিদ তুমি তো আলোক ময়দার ল্যাপটপের সামনে বসে আছো তুমি একবার আলোক ময়দার ল্যাপটপের সামনে যাও আমি বিসিকিউ হল থেকে একটু কথা বলছি পোডিয়াম মাইকে দেখো তো শুনতে পাও কিনা আচ্ছা আপনাদের কেউ এখানে জয়েন আছে একটু বাইরে গিয়ে দেখুন না আপনাদের সাথে কথা বললে হয় না না যারা জয়েন আছে
হ্যালো হ্যালো
বাইরে নিয়ে যাও, ওটাকে বাইরে নিয়ে যাও, ওই ফোনটাকে বাইরে নিয়ে যাও। হ্যালো, হ্যাঁ, তুমি কি ল্যাপটপের সামনে একটু আনমিউট করো, একটু আনমিউট করো। আচ্ছা, তুমি এবার গুগল মিট লিংক থেকে দেখো শুনতে পাও কিনা। আমি ফোনটা কাটছি, ফোনটা কেটে দিচ্ছি আমি। হ্যালো 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 হ্যালো, এটা কেন হচ্ছে বলো তো? ও দাঁড়াও তো, সিস্টেম স্পিকার একবার দেখবো, ট্রাই করবো। হ্যালো, শুনতে পাচ্ছ নাহিদ? নাহিদ শুনতে পাচ্ছ? নাহিদ শুনতে পাচ্ছ? ফাইন ফাইন, ঠিক আছে ঠিক আছে ঠিক আছে ঠিক আছে। সিস্টেম স্পিকার, হ্যাঁ, ডিজিটাল মাইক্রোফোন না, সিস্টেম স্পিকার দেওয়া আছে, ঠিক আছে। নাহিদ তুমি আরেকবার থাকো, আমি আনমিউট করে মিউট করছি, তারপরেও একবার দেখে নাও শোনা যাচ্ছে কিনা। আনমিউট করো। হ্যাঁ নাহিদ, এবার শুনতে পাচ্ছ? ঠিক আছে, থ্যাংক ইউ থ্যাংক ইউ থ্যাংক ইউ। ওটা ডিজিটালটাই থাক, সিস্টেম ডিজিটাল স্পিকার, সেটা স্বাভাবিক। আচ্ছা, এইবার কি করবেন? আচ্ছা, নেক্সট স্পিকার হচ্ছে, ও একটা জিনিস দেখতে হবে, একটু ছাড়বেন। আচ্ছা করুন। ম্যাম কোথায়? শোনা যাচ্ছে শোনা যাচ্ছে। হ্যালো হ্যালো হ্যালো, নাহিদ শুনতে পাচ্ছ কিনা দেখো তো। হ্যালো,
হ্যাঁ শুনতে পাচ্ছি।
ঠিক আছে। কিন্তু এইটা প্রেজেন্ট হবে। আচ্ছা, গুগল মিট থেকে এখানে লগ আউট করে দিচ্ছি তাহলে। আচ্ছা ম্যাম, ও লাগছে না গুগল মিট।
হ্যালো হ্যালো,
Hello.
Hello. Hello.
Hello.
Hello.
Hello. Hello.
Please, who are outside, please take your seats. The session will
Hello.
start now.
Hello.
Please take your seats. Huh. Please take your seats, sir. Please take your seats. The session will start.
Hello. Hello.
তারপরে আমি আপনাকে বলবো আগে প্রফেসর তারপর প্রফেসর উন্নত না তারপরে হ্যাঁ উন্নত তারপরে না ওইটাই চেঞ্জ হচ্ছে ওটাই হ্যাঁ প্রফেসর আটো একটু আগে বলতে চেয়েছি না তারপরে হ্যাঁ তারপরে উনারই ছিল হ্যাঁ উনারই ছিল না আমি অদিতি বেরিয়ে গেছে অলরেডি All of you, please take your seats. We are going to start our session. আচ্ছা আমি একটু স্টার্ট করছি তারপরে ডাকছি as we all know that today we have assembled here for the centenary celebration of Professor J.J. Ghosh through an international seminar on biochemistry then and now, jointly organized by Professor J.J. Ghosh Foundation and Department of Biochemistry, Calcutta University. Already we have the inaugural speech by Professor Shankar Mitra from USA, and we have the lecture of Professor Shankar Addo. You can imagine that it is 11 p.m. in USA. So we have to conclude this part as early as possible. And now we are going to start our formal technical session, offline technical session today. And there are many lectures here. We have Professor Arun Dotto, Professor Dhrubajyoti Chattopadhyay before lunch, and after the lunch we have Professor Gauri Shankar Ghosh. So now we are going to start, but this will be first I shall invite Professor Shanghamitra Sengupta, Head of the Department of Biochemistry, Calcutta University, to deliver her welcome address. Madam, please.
Good morning. Please settle in your seats, dear students. Yeah, please come back and settle in your seats. So this is an formal welcome address. Also, the seminar has started almost an hour and a half before. Okay, good morning, distinguished guests, esteemed colleagues, and my dear research scholar and students. Please settle down. It is my utmost pleasure and privilege to warmly welcome you all on behalf of the Department of Biochemistry, University of Calcutta, to this much anticipated one-day international seminar entitled Biochemistry Then and Now. This event is a heartfelt tribute to commemorate the birth centenary of the legendary educator and scientist Professor Jagat Jiban Ghosh. For over almost 6 decades, maybe little more than 6 decades, this department, the Department of Biochemistry, has stood as a beacon of scientific excellence, nurturing brilliant minds and fostering groundbreaking research. Professor Ghosh was an exemplary teacher and an outstanding scientist who played a monumental role in shaping this department as well as the biochemistry education in the country. Therefore, hosting this event is a true honor for our department as we bring together renowned experts from around the globe alongside passionate learners and researchers in biochemistry and related fields. So, dear students and scholars, I assure you that today will be an inspiring journey for you. You will witness transformative discoveries, thought-provoking presentations, and stimulating discussion. And therefore, and last but not the least, I would like to extend my heartfelt gratitude to the dedicated organizing committee, our scholars, and Jagat Jiban Ghosh Foundation, whose tireless efforts and contributions have made this event possible. My heartfelt gratitude to all of you. So without further ado, I once again welcome you all and wish you a highly engaging and rewarding experience. Please enjoy. Thank you.
Thank you, madam, for your wonderful deliberations. Now, I shall invite Professor Mrinal Kanti Poddar, President, Professor Jagat Jiban Ghosh Foundation. He was the Emeritus Professor, Department of Biochemistry, Calcutta University, also Emeritus Medical Scientist, Jadavpur University after retirement. So, sir, please deliver your lecture.
Thank you. Oh, এটা চালু হবে কারণ এইটা দেওয়া হবে না হ্যালো হ্যালো ওকে any problem okay very good gathering huh? Good morning and very happy new year 2025. Our today's honorable speakers, delegates, teachers of the biochemistry department, our scholars, students, and my friends. It's a pleasure and privilege for me on behalf of the foundation and myself to welcome you all in this August gathering of our international hybrid program in the occasion of centennial celebration of our beloved eminent teacher, Professor Jagat Jiban Ghosh. Professor Ghosh was an erudite scholar and a legendary teacher of teachers and scientists all over India and abroad in the field of biological sciences. Professor Ghosh's dedication was enormous, and his contributions were incomparable in the development of biological sciences, especially biochemistry, neurobiochemistry, and its allied fields, even in the biomathematics. In those days in India, particularly in the then Calcutta. He had been associated with teaching and research activities of these universities as well as other universities, institutions, hospitals, industries, even in a blind school, Calcutta Blind School, which is situated at Behala. To honor such a knowledge-attractive, intuitive, dynamic, brain-mind-induced, vibrating as well as inspiring personality, Professor Jagat Jiban Ghosh. This foundation was established in the year 2012 by his students, colleagues, friends, and admirers' generous contributions for the fulfillment of the J.J. Ghosh's commitments to education and research for the students. To fulfill his mission and visions, this foundation started and continuing since 2012 its yearly activities such as birthday memorial lectures, winter special memorial lecture, World Health Day lecture for the schools students. And this foundation also organized for the undergraduate students the national and international seminars. And this foundation also organized for the postgraduate biological sciences students a critical essay writing competition. In addition, this foundation is also given an award to the MSc Biochemistry students of Calcutta University every year who secures the highest marks in Neurobiochemistry in their final exam. In fact, altogether, I think this foundation so far organized till now 45, 46 or a little bit more events during these last 12 years. And for the benefit of the students of the schools, college, as well as the universities. Last year, on July, our foundation, in collaboration with the Department of Biochemistry, University of Calcutta, has undertaken a year-long celebration of centennial of Professor J.J. Ghosh. And during the last few months, scientific lectures for undergraduate students were held at Vidyasagar College on August 29th of the last year, at Vivekananda College at September 30th, as well as at Ashutosh College on November 30th. In addition, for the postgraduate students, a biological of the biological scientific streams, critical writing competition on microRNA role in was held online from November 14 to 17 last year. Today, on the occasion of the centennial celebration of Professor J.J. Ghosh, a one-day international hybrid seminar on biochemistry then and now is organized. And in addition, a museum in the memory of Professor J.J. Ghosh will be inaugurated in this department in his memorial hall by Dr. P.K. Sarkar, the former INSA scientist of the IICB, in the Indian Institute of Chemical Biology, Calcutta. With this brief introduction about the Professor J.J. Ghosh Foundation, on behalf of myself and on behalf of the foundation, thanks to everyone for your presence in this August gathering to grace the occasion and make our celebration program a grand success. Our thanks are also to the donors of the generous contributions to support the foundation. Once again, on behalf of the foundation and myself, our heartiest welcome to you all. Thank you.
Thank you, sir, for your motivational speech. Now I shall call upon Dr. Aditi Nag Chaudhary to introduce Professor N.C. Mondal, Chairman of the technical session today. Second, it is noteworthy to mention that second Professor J.J. Ghosh Memorial Lecture was delivered by Professor N.C. Mondal in 2013. So, Madam.
Coming from a remote village of Midnapur, he has left the village and came to our Department of Biochemistry and did his PhD from Professor B.B. Bishas of Bosch Institute, and the research was in plant biochemistry. Professor N.C. Mundol did postdoctoral research in USA at the Department of Molecular Biology, Albert Einstein College of Medicine, New York, with Professor J.T. August from March 1970 to 71 on RNA bacteriophage. Then he shifted to Professor Margaret Lieb from January 1972 to 73 and worked on temperate bacteriophage lambda. Then he joined Bosch Institute as a lecturer and retired as professor and emeritus scientist in 2003. In his independent research career, he developed through self-taught manner the full-fledged molecular genetics using bacterial and viral systems, coliphage lambda, mycobacteria, and temperate mycobacteriophage, L1, to understand various aspects of gene regulation in those systems at molecular level. His papers, original research papers, were published in Journal of Biochemistry, Virology, Biochemistry, Journal of General Virology, Proceedings of National Academy of Science, Genes and Development, Microbiology, Molecular and General Genomics, Journal of Biochemistry and Molecular Biology, Protein Engineering, etc. All the journals are of high impact factor. He has got a number of awards and recognitions. Of those, I will mention few. Gold medal from the Calcutta University for standing first class first in MSc exam in 1962. Shoilendra Sarkar Memorial Lecture Award from Calcutta University in 1997. Professor J.J. Ghosh Excellence in Teaching and Research Award for 2012 from Calcutta University. Shardar Patel Award 2013 for Excellence in Youth Development from V. Patel Foundation, New Delhi. Bharat Ratna Mother Teresa Gold Medal Award from Global Economic Progress and Research Association, New Delhi. Obviously, such a person, such a colorful person, should be a member of a number of learned societies, and in all the societies he was elected member. Fellow of Indian Academy of Science, Bangalore, Fellow of Indian National Science Academy, New Delhi, Fellow of National Academy of Science, India, Allahabad, Fellow of the Indian Chemical Society, member of Guho Research Conference, life member of the Society of Biological Chemists. He taught molecular genetics, molecular biology, virology to the PG students of biochemistry, microbiology, biotechnology of Calcutta University, Kolkata University, Jadavpur University, Vidyasagar University, IISER, and also at Lady Brabourne College. Served as a member of various national committees relating to academic affairs. By invitation, he wrote various articles on popular journals and newspapers. He authored a number of books, and I just mentioned book on Acharya Jagadish Chandra Bose, on Acharya Prafullo Chandra Rai, on Vidyasagar, on History of Science in India, the part volume 8, the biochemistry, biophysics and molecular biology part was, he authored with a co-author, Professor Dhrubajyoti Chattopadhyay, which was published by joint venture by National Academy of Science, India, and the Ramakrishna Mission Institute of Culture, Gold Park. He also authored books on coronavirus 2019, The Twilight Years, Understanding Human Aging and Death at Molecular Level, published by Nammo Press, New Delhi. It is also available from Amazon, India. He has guided 18 PhD students. All of them are in responsible public services at all places in different capacities. His other activities, he was engaged in national science movement. During past 15 years, he extended financial help in form of donation or otherwise to different organizations for the development of science. Even at schools, he has given a lot of donation. His own village school, he has donated books of rupees 1 lakhs, and he also helped other students, other schools. He has actually taken a mission to make scientific mind from the school level, from remote village to urban students till PG. He has donated a large amount to Professor Jagat Jiban Ghosh Foundation, Department of Biochemistry. He has talked on radio. He has written a number of popular articles, and actually it will take huge time to say everything of this colorful man. So I must say that Professor Ghosh, be with us, Professor Mondal, be with us till you on this earth. Thank you.
Thank you, Madam, for nicely introducing Professor N.C. Mondal before the August gathering. Now, I shall call upon Professor N.C. Mondal to introduce our third speaker, Professor Arun Dotto. Professor Arun Dotto. Professor Arun Dotto. Professor Arun Dotto, I would just want to say that Professor Arun Dotto has been the judge of critical essay writing competition held by the foundation every year. And besides being the judge, he also gave online suggestions to the students about doing the reference work during the critical essay writing. And also, you will be very happy to know that Professor Arun Dotto have done PhD. It's submitted his thesis under Professor Jagat Ghosh. So, sir, please introduce.
It is my pleasure to introduce some of my students here on this dais. Dr. Arun Dotto got his BSc honors in chemistry from Surendranath College, Calcutta in 1973, and MSc in Biochemistry in 1975, and PhD in Biochemistry in 1987 under the guidance of Professor Jagat Ghosh and Dr. V.M. Kattach, while working as a scientific staff member at the Jalma Institute of ICMR at Agra. In 1985, he was nominated for British Council Fellowship by the ICMR and spent 6 months at the University of Newcastle upon Tyne, UK. He had PhD training at University of Wisconsin-Madison from 1987 to 1990. Then he joined as an instructor at University of Texas Southern Medical Center at Dallas. Later, he moved to San Diego, California, to take a scientific staff position, a joint position with Cytel Corporation and the Scripps Research Institute. In 2004, he joined the National University in San Diego, California as a full-time faculty member and served as a director of one of the centers. With his academic background and expertise in both molecular biology and computer science, his research interests included glycobioinformatics and health informatics. His team developed multiple software tools used by various organizations, including the Illinois Department of Public Health for their childhood obesity control program. Currently, he teaches computer science as a part-time professor at National University and serves as a consultant for various companies and organizations. Actually, এটা পাস করার পরে তো আমি বসে ছুটে আছি আর এখানে চলে গেছে। এদের সঙ্গে অরুণের সঙ্গে পরিচয় হলো ওই করোনার পরে যে রবিবাসেরই একটা গ্রুপ কাজ করতো, সেই গ্রুপের সঙ্গে গিয়ে অরুণের সঙ্গে পরিচয় হলো। যাই হোক, অরুণ পরে আমার বাড়িতে এসেছে, ওর বাড়িতে গেছি, ফোনে কথা হয়। যাই হোক, I invite Dr. Datta to deliver his lecture.
ফুল মোডে দিয়ে দে এই সে ও আচ্ছা আচ্ছা এখানে আছে
ও কি বাইরে যাচ্ছে, লিংকে যাচ্ছে।
So biochemistry then and now. The impact of the computer science in biochemistry. Can everybody hear me? Yes. Okay. Okay, maybe I can take that. Can you hear me now? Yes. Okay, thank you. So the topic, of course, is the biochemistry then and now. So everybody, those who are students here in biochemistry, they know the advancement that is going on in the biochemistry. But the impact of computer science in the biochemistry, today I will actually introduce you. So you'll see that how the computer science is making a depth in the advancement of biochemistry, not only biochemistry, but all other sciences. And I'll just briefly touch about this. So the title is Generative AI, a paradigm shift in search function for education and research. I don't know how many of you are using generative AI for everyday use. Of course, everybody uses Google search. Google search gives a list of all the
different list of all the hyperlinks. But what generative AI does, once you put the prompt, is called prompt engineering. Once you put the prompt, you get a chat-like output. And it's very much useful now for most of the people who are doing the research using the generative AI. So this is very appropriate in case of Professor Jagat Ghosh's methodology of teaching. The term is called pedagogy, a kind of scientific procedure how you can create an environment with the students so that the professor can provide the information that he likes to do with the students. And of course, most of us, those who are already aged, we got a chance of Professor Jagat Ghosh's talk, and he was a very well speaker. I don't know whether he really did any work on the pedagogy, but he must have done whatever he knows about the pedagogy. So the term pedagogy refers to the method and practice of teaching which encompasses the strategies. Can you turn off this one, because this is working, otherwise there is an interference. And approaches yes. And it's not just about the act of teaching itself, but also involves understanding how students learn and how to create effective learning environments. Those who got a chance of taking the class from Professor Jagat Ghosh, everybody knows that how he actually created the atmosphere, the environment for reaching out to the students, understanding the very difficult subject, molecular biology, genetics, and neurobiology. So in National University, I was doing some research with some other professors, and some of the publications I have given here. Let's go through this. What is called generative AI? I don't know how many of you are using generative AI. I'll go through that very briefly because the time is very short. So how the generative AI actually evolved, what is called generative AI, what kind of impact generative AI is having in research and education, that I'll briefly go through. So the generative AI, or simply termed Gen AI, is a disruptive technology. Probably all of you know that once the internet actually came in our life, daily life, we are using internet every day. Believe me, generative AI is going to change the way the internet changed you. So we'll be using, especially the new generation of students, you'll be using generative AI every day for your research and education. So this particular technology has already impacted almost every field of like education and research that I'll be talking about and focusing on that part. Literature, art, finance, advertising, tourism, just to name a few. So the Gen AI is a subset of artificial intelligence that creates new content such as text, images, videos, audios, based on input prompts. Prompts is nothing but, a paragraph. Say, for instance, you want to know something about virology, what Professor N.C. Mondal has given and gave you the introduction. So if you give one paragraph of virology, you like to know more about it, it will give you almost one page of text output showing what it means, what are the different divisions of virology, and so on. It uses large language models which learn patterns and structures from existing data to produce new data. Example, ChatGPT, most of you might have already heard, is from the OpenAI, Gemini from Google, and Copilot from Microsoft. These are some of the examples. So why generative AI? Why the term is coming? Because it generates new content. For example, a generative AI model trained on a large data set of images can create entirely new images that look similar to those in the data set. Similarly, a model trained on text can generate new sentences or paragraphs that mimic the style and content of the training text. These are the different, this is a simplified overview of the process the generative AI actually creates. The text is a data collection, then training, then learning the patterns. Generative AI has to learn the patterns. Generation, fine-tuning. So these are very shortly I actually mentioned, but if any one of you want to know more about it in depth, I can have after the talk at some time. You can approach me, I can explain further. So the term artificial intelligence is a generative AI is a subset of artificial intelligence, was coined by John McCarthy in 1956. Is often referred to as one of the founding fathers of AI. So the concept of generative models has been around for decades with the early examples like the ELIZA chatbot created by Joseph Weizenbaum in 1961. And then, of course, in 2014, everything changed with the advancement of the deep learning in the field. In 50s, the concept of machine learning began with Arthur Samuel. In 1960s, Joseph Weizenbaum created ELIZA. I'm just briefly giving you some interesting background. And then the neural network is actually part of the neural network, is a backpropagation algorithm. Then 80s, 90s, neural network gained popularity, and researchers developed more sophisticated models and algorithms. So in 2000, introduction of variational autoencoders and the resurgence of interest in neural networks see the stage of more advanced generative. In 2014, Ian Goodfellow and his colleagues introduced generative adversarial networks, which significantly advanced the field by enabling the generation of highly realistic images. So in modern era, OpenAI released the first version of GPT in 2015. In 2020, the development of models like GPT-3, DALL-E, and other transformer-based architecture. What it is, I'm going to give you a very short overview. Further pushed the boundaries of what generative AI could achieve, including text, image, and even video generation. So transformer models was a part of neural network. It was introduced by Fashwani et al. In 2017. So he actually showed by the transformer model that the whatever could not be achieved by simple neural network could be achieved by this transformer model technology. What it does, it actually enhances the neural network technology and does a massively parallel algorithm, the computational algorithm, to provide the text in number of areas. It also, instead of one single attention mechanism, it uses multiple attention heads. Each head operates independently and focuses on different parts of the input sequences. So when it does, it actually uses the token. Token is equivalent of the computer for the word. So say, for instance, hello is a word. So that is used by the generative AI in terms of the token. It actually breaks it down. So around 750 words is equivalent to 1,000 tokens. And Gemini is using almost like 1.5 trillion parameters. So you can imagine that how powerful and how many people, researcher, high-level researcher, both in mathematics as well as in computer science, are involved developing this Gemini. My understanding is Dr. Shumantada's son is also involved in one of those projects. Everybody knew in 2024, it has been awarded is the year of artificial intelligence. It was awarded to Hopfield and Geoffrey Hinton because they are the pioneer of the discovery of the neural networks, the artificial intelligence. And this is one publication I just quoted. If you are interested, you can actually see that deep learning for artificial intelligence has been published in the Communication of Computer. Then in the chemistry, it is the David Baker, Demis Hassabis, and John Jumper. David Baker is in the University of Washington, and Demis Hassabis and John Jumper, they are in the DeepMind in the Google who actually developed the technology. They developed what is called AlphaFold 3, and David Baker developed what is known as Rosetta. What is Rosetta? What is AlphaFold 3? Let me give you a very short, brief introduction. So David Baker actually did a groundbreaking technology. So you can, using David Baker's algorithm, Rosetta 3 for example, to create an enzyme which doesn't exist in nature. And the impact is now in the pharmaceutical industry, there is a, I'll come back to that one. Pharmaceutical industry, they like to synthesize the molecules, especially the glycan molecule, by using chemoenzymatic method, which is much more direct. But if the enzyme is not there in nature, how can you do it? With the David Baker's technology, you can develop, you can create the enzyme. That's why he was given the Nobel Prize. A lot of people I know, they are actually collaborating with him, including the pharmaceutical industry. Another groundbreaking is from the DeepMind of Google by Demis Hassabis and John Jumper. And what they did is all the proteins known, almost like 1 million proteins from bacteria, virus, smaller organism, all proteins, predicted structures are in their server. All proteins predicted structure in the server. So if you want to know what might be the possible structure of your protein of interest, you just go to the NCBI's website, cut and paste the FASTA and put it in their server, and you will see the structure. And it is in their AlphaFold 3, the structure prediction has become so accurate, almost like more than 99%, that a lot of pharmaceutical companies just name it, they are collaborating with them. So they have actually started a company. Thank you. So they recently, Hassabis, he is from UK. So he actually started a company called Isomorphic Labs. And if you go to the Isomorphic Labs website, you can see what they are doing it. It's mind-boggling. So why I'm presenting all this? Because the impact of this technology is so profounding in the biochemistry and molecular biology that from now on, without this kind of technology, it cannot progress the way it is progressing now. So these are some of the publications I had. My expertise, not only in the computer science, but also in molecular biology. I did the structure function analysis of a kind of enzymes that actually create the glycan. Glycans is defined by the monosaccharides. And what it does, because the glycan is all over the cells, outer cell membrane, then the interaction between two cells actually is caused through the glycans. So I was doing all those structure function analysis of the glycan related molecule. That drew me the attention that maybe we know the code from the sequence of the nucleic acid, the sequence from the protein, but the sequence of the glycan is very difficult because it doesn't actually use the central dogma. Each and every monosaccharide is attached to another monosaccharide with a very orchestrated fashion by the glycosyl transperases and the glycosidases. So I have some review article I did not present here, but some of you actually can go through that. So with this in mind, what I'm doing now is focusing, not only I'm doing, Caroline Bartholdi, who actually got Nobel Prize in a couple of years ago, maybe 2 or 3 years ago, and she is a pioneer, is the click chemistry for synthesizing the glycan. So a lot of people are now focusing on the glycocode. In fact, I wrote an article for a book on the glycocode. What are the different technologies that are needed for the deciphering the information that are remaining in the glycocode? So what it says, surface of all living cells decorated with glycan molecules that are conjugated with proteins or lipids play significant roles in cell-cell communication, among others. Glycans, more specifically glycocodes, which are complex carbohydrate structures consisting of 10 monosaccharides in humans, encode information for various biological processes. So glycomics workbench we are developing, designing and developing for the better understanding of every glycan molecules. And I have a demo. I don't want to start now. I'll show you at the end. So now coming back to the impact of generative AI on education. Now some of you are teaching in this department. So what the advantages of generative AI in education is a personalized learning. And in US, a lot of students actually do the homeschooling. So for them, generative AI has become a very nice tool because it's actually helping them understanding the science through personalized learning. Automated administrative tasks for the educators, enhanced student engagement, support for diverse learning needs, efficient content creation, immediate feedback, research and analysis. So if a student writes a research article and you put it in the generative AI as a prompt engineering, as I mentioned, then you can actually get the feedback of your research article. So I suggest that everybody starts using generative AI, you will see that how powerful it is. And these are some of the explanation what is meant by personalized learning. I am not going through this. Automated administrative task, teachers can save time by using GenAI. Enhanced student engagement. Everybody knows how Professor Jagat Ghosh actually used to do that. He engaged the students, and generative AI actually can help. He would have loved it. He would have loved it. Support for diverse learning needs, efficient content creation, immediate feedback, research and analysis. But it cut in both worlds. It has also disadvantages like lack of empathy, over-reliance, bias and inaccuracy, limited creativity, distraction, ethical concern is very important. Now, almost every funding agency like NIH, NSF, they are funding for the ethical use of AI-generated data, and is a resource intensive. So how much resource is needed for developing a tool like generative AI? I'll briefly touch base on this. I'm not going through this definition of over-reliance, bias, limited. Okay. So now, if you want to run a tool like Gemini or Copilot, then what you need? You need high performance GPUs, graphical processing unit, like CPU is a GPU. It has a high memory requirements. It has a high energy consumption, so you need cooling and power delivery. You have to have infrastructure, not only the physical infrastructure like hardware and software, but also the people who can run it. And there is not many people who are trained in AI, so there is a big demand in US. So if you know how to run it, then you have a job. Overall, the computational power needed for GenAI is immense, involving advanced hardware, significant energy resources, and sophisticated infrastructure. So that's why very limited number of companies actually are providing you that kind of tool. And once the generative AI came into the market, there is almost like hundreds of companies grew up, but they are at the back end using this kind of tools. So for the glycomics workbench, unfortunately, I cannot use the commercial source, not only for the cost involved, but also the researcher, they actually need some sort of a full control, confidentiality, and the security. So what we are doing is we are using the Llama 3 from Meta, and National Science Foundation has an infrastructure called Access. So plan is to put the Llama 3 there and then train it. Access will provide the infrastructure for running that kind of tool. So it's not only the system requirement if you want to do it. Choosing an AI model, we are using the Llama 3. Data acquisition and preparation, my work is totally focused on the glycobiology for defining all the glycan molecules. And for training, data acquisition and preparation, you need what is called retrieval augmented generation frameworks. And what it is that, if anybody wants to know, I'll be outside, you can come back and can ask me. Then you have to train the model, and you have to estimate the resources that you have. You have to design the system, deployment, and monitoring. Of course, for monitoring, you need a set of people highly skilled. As I mentioned, if you know how to run generative AI models, you have a job immediately, even if you don't have the degree. Maintenance and updates. And implementing a GenAI system is a complex process that requires careful planning and execution. And so what we are planning to do is developing an application programming interface so that the users in the glycomics workbench, they can actually utilize the Llama 3 in Access. So these are already I have covered. Literature review, hypothesis generation, that's very important. Gone are the days when we used to go to the library, 70% of our time was one in the library while we did the work and doing the PhD. But now with the internet, we can do it from home, and an extensive amount of time is saved because of the internet. What the generative AI is going to do is they will actually create the materials for you, writing materials. Of course, you have to go through that, make sure that all the informations are correct, because one of the defect of the generative AI is the hallucination. That means the output sometimes is not the real. But in science, especially in biological science, we have the advantage that we can go to PubMed and can verify that whatever it is given, whether it is right or wrong. Efficiency, productivity, innovation, because once you learn more and more, you become more innovative. And disadvantage is digital divide. Some people will not be using, so that will create a digital divide. So ongoing developments, quantum computing is also going to change the way the generative AI is used. I think some of you have heard the quantum computing. And then instead of Python, which is used for developing these generative AI tools, maybe Julia can use, which runs almost 10 times faster than Python. So those are the investment, ongoing developments are taking place. And of course, it is the policy and the regulation that will regulate how generative AI can be used. So in conclusion, everybody, students and educators both, can be benefited using this particular technology. Benefits outweigh the risk involved. Policy should focus on the benefits while mitigating the risk. Cost to run a customized system or pre-trained system is a major factor to be considered. And the rapid development of this technology can reduce the cost and democratize the usage, particularly by the educational organizations. With this, I'd like to stop here. And if I have another 1 minute time, I can show the demo. Do I have?
Yes, sir.
Okay. So one thing because I'm using another computer, for using the generative AI, you need to have an account. And I have an account with the Microsoft through my university. And unfortunately, I did not bring my US phone with me. So to access to my account with the university, I need a code. And that code I cannot provide. So what I'm showing to do it now. So anybody can give me this should not be the way. Just another couple of seconds. If it doesn't work, then this will be my end of the talk. ইন্টারনেটটা চলছে তো? ও আচ্ছা আচ্ছা। So this is the portal we are developing. It will take a long time if I want to show each and everything, but what I like to show you very glimpse what it means. So it actually shows what the about is working. What is large language model? If you want to use, you can. What is glycans? What are glycocodes? There are 10 monosaccharides which actually creates the glycocode in the human that are responsible for all the interaction that occurs. And one of these is the sialic acid. Oh, sorry. Anyway, I think I have taken enough time, so yeah. So these are the monosaccharides which actually consist of the glycocodes. And you can see the definition of all those glycan drugs and everything will be here. One thing is, so one you have, I actually wrote an article for the NCBI almost 25 years ago on a molecule called ST6-GAL1. And since then, a lot of changes have taken place. And so these are so one can actually define each and every molecule with all those informations. And once it is there, then you get details about it. So with this, I'm ending here, and I'm really grateful for giving me the opportunity to talk about it. Thank you.
Thank you, sir. They will be felicitated, Professor Arun Dotto will be felicitated by Professor N.C. Mondol with a memento and a potted plant. So, Professor Arun Dotto will be felicitated on behalf of Foundation and University of Calcutta Department of Biochemistry with a memento and a potted plant.
I think this will be difficult to take it to US. Okay. Thank you, sir.
Thank you, sir, for delivering such a lecture before the August gathering. Now, Professor N.C. Mondol will now introduce Professor Dhrupajyoti Chattopadhyay, our invited speaker. I want to mention that Professor Chattopadhyay did his PhD under Professor N.C. Mondol. And Professor Chattopadhyay also delivered the 12th Professor J.J. Ghosh Memorial Lecture. There he showed us the class notes used, class notes of Professor J.J. Ghosh from a khata which he preserved till now. And Foundation has asked to this khata to keep in the as an exhibit in the Professor J.J. Ghosh Memorial Hall. May we get that khata, sir, from you? Yes, we are. He has already given us the permission to display that khata in the exhibit, as the exhibit. So with this, I give the microphone to Professor Mondol.
Professor D. Chattopadhyay needs no introduction here because he is well known. Only just কিছু বলার আছে অনেক কিছু যেগুলো জানি না কিন্তু এখানে সময়টা কমে প্রেসার আসছে যে যাতে না বলি এনিওয়ে ও ডক্টর আমি বাংলাতে বলছি তাড়াতাড়ি হয়ে যাবে প্রেসিডেন্সি কলেজ থেকে বিএসসি অনার্স কেমিস্ট্রি পাস করেছে বোধহয় 73 এমএসসি ক্যালকাটা ইউনিভার্সিটি 75 ফার্স্ট ক্লাস সেকেন্ড সমিত আড্ড বোধহয় ফার্স্ট ছিল তারপরে হি জয়েন মাই ল্যাবরেটরি কিন্তু পিএইচডি শেষ করার আগেই ও আশুতোষ কলেজে জয়েন করেছিল সেখান থেকে ক্যালকাটা ইউনিভার্সিটিতে এসেছে বায়োকেমিস্ট্রি ডিপার্টমেন্টে তারপরে থিসিসটা জমা দিয়েছিল এনিওয়ে যাই হোক এরপরে অনেক কিছু তার উত্তরণ হয়েছে সেটা সিনেটের মেম্বার মানে ডিন হয়েছে ক্যালকাটা ইউনিভার্সিটির প্রভিসি হয়েছে একাডেমি ক্যালকাটা ইউনিভার্সিটির এমিটি ইউনিভার্সিটির ফাউন্ডার ভিসি নাও নিবেদিতা ইউনিভার্সিটির ভিসি তাছাড়া হি হ্যাজ ওয়ার্ল্ড লাইক ট্রাভেল সো মেনি ট্রাভেলস টু ইয়োর his laboratory tour etc. যাই হোক এই বলে I am proud of him as his mentor for PhD. Okay. Please come.
Again.
Very good afternoon to everybody. At the onset, I just like to thank Professor Meenal Poddar, the president of this J.J. Ghosh Foundation, and all the members of the J.J. Ghosh Foundation, and my colleagues from the Department of Biochemistry, my dear students and research scholars who are present over here, and also my other friends who are present here. When actually Aditi Nag Chaudhuri approached me that you have to deliver the lecture in this conference after this stalwarts in the biochemistry, I was thinking that what should I do? I thought that why not I try to give some idea about what is biochemistry from the very beginning and till when Professor Ghosh actually joined us, started this department. Then I was thinking, is that going to be a little bit not right for me to do justice to this particular offer. So I thought that I shall try to give you some idea to justice the title of this lecture for this conference theme. What is that? It is the biochemistry, then and now. এটাকে connect করে দিতে yeah. Okay, so this is what I thought that I shall do, but you can easily understand this is a huge topic. I shall not take much time, but I shall try to give you an overview, because what I learned from my sir, the teacher of teachers, Professor Jagat Jiban Ghosh, that if you are a student of biochemistry, you have to update yourself continuously. You have to learn every day. Almost you can see that while we are teaching biochemistry, sir used to come with the papers. Dhruvo, I got this paper. This is very important. You can read it and then share it with your students. That's what is Professor Jagat Jiban Ghosh. And from there, if I can actually yeah. So what I thought that trying to understand the life, what Sad used to say that if you look at the life in the plants, to the animals, to the humans, so the vegetative soul is in the plants, and then we have the animals where we have the sensitive soul, and then we have the humans which are belonging to the animals, but where the rational soul came. And all these things are actually related to the couple of things, what we can say as reproduction, growth, mobility, sensation, thought and reflection, what we could find in the animals. So the Aristotle classification of the soul with fixed species and a short biblical time scale is that one. Now in geology, the evolutionary time scale slowly emerged from the tools. And if you look at this, actually this is actually how we can look at the development of this whole generation and trying to understand life. Why I'm mentioning this slide? This is very important. In our first class, Professor Ghosh actually mentioned to us that how we can define biochemistry. Bios means life, so chemistry of life is biochemistry. And that's why I thought that I should start my slides with the life. Now, biology gradually became evolutionary, culminating in Darwin's Origin of Species and Descent of Man. And over here, what we can see in 1859, when the origin of species, that is, populations evolve over the course of generation through a process of natural selection. But if we look at 1871, the descent of man involved over millions of years from the African apes. So these are the two where we can get the background of the evolutionary participations. But there are different views of life. Say, for example, vitalism. And vitalism is a belief which ultimately descended from Aristotle's theory of soul and is based on the premise that living organisms are fundamentally different from non-living entities because they contain some non-physical element or are governed by different principles than are inanimate things. But if you look at the mechanistic view, in the mechanistic view, it proposes that the entire universe, including living bodies, are controlled only by mechanical forces. But so for example, the very first example, what we can get it from the synthesis of urea in the early 19th century by Frederick Euler from inorganic compounds was counter evidence for the vitalist hypothesis that only organisms could make the components of living things. And that's why we can see the idea of separate laws which govern the inanimate and the animate was threatened by the chemical revolution, in particular by Lavoisier's development of the analogy between combustion and respiration. Now, also his law of the conservation of matter arose from his study of fermentation. Chemistry and biological processes were becoming intertwined. And this is one of the very favorite subject area of Professor Jagat Ghosh. And that's why while in the classes, after the lectures and other, he actually tried in our reunion to have one particular theater play on Louis Pasteur. The Louis Pasteur actually, how this, and he was so passionate actually to have that play to be in the reunion, but we can't actually. In the 19th century, chemistry in its biological aspects gradually achieved a consistently materialistic approach. And here, the foundations of biochemistry, if we look at it, which comes during the 19th century organic chemistry, founded on the enormous advances in organic chemistry, together with elucidation of the nature of the cell, laid the foundations for the development of biochemistry as a separate discipline. Now, the two critical developments as early as the 1830s were the discovery of isomerism, which get the first hints of the complexity of organic molecules, and the identifications of enzymes as catalysts. However, it was around the turn of the century that the behavior of enzymes really began to be elucidated, and with the establishment of the composition of simple proteins, biochemistry developed in earnest. So this is, I am sure that all of you know about this, the enzyme, the active sites, substrates, which are there. Now, between 1810 to 1830s, if you look at, we can see the isolation of substances composed of carbon, hydrogen, oxygen, nitrogen from both plants and animals. The term protein, most important thing, was first used in 1838. But if you look at the next generation from 1850 to 1890, the carbohydrates, lipids, nucleic acids were identified, and in 1870, the term biochemistry was first coined. Now, when this term actually coined, the biochemistry, understanding the scientific basis of life. So here, I just want to mention about one of the very, very interesting quote from J.B.S. Halden in his Mechanism, Life and Personality in 1913. We must find a different theoretical basis of biology based on the observation that all the phenomena concerned tend towards being as coordinated that they can, what is normal for an adult organism. Now, here, this particular statement, they express what is normal for an adult organism. So, the ideological problem for biochemistry was to remove in all its aspects vitalism. If you remember, then the slide, the vitalism is the one what is proposed first, or the idea that life depended on a special vital force. So, in the 20th century, we could find that there are technological advances in the early 20th century, like the X-ray crystallography, the electron microscopes, and all this actually helps quite a bit. Now, if you actually consider this particular very important publication, What is Life? Very gradually, the giant biological molecules are coming to be understood as aperiodic polymers, culminating later in Erwin Schrödinger's seminal book, What is Life? So this is a book, Schrödinger's book. I really want that those who are interested, please go through that book, because the concept, how it developed, is something which is really fascinating. And then definitely I should mention about Perroud's, with his hemoglobin molecule in 1950s. The structural biochemistry gave valuable insights into functions and mechanisms of biochemical processes. Say for the extra structure of a protein myoglobin at 6 Armstrong resolution, or the model of the catalytic triad and oxyanion hole of chymotrypsin. This is also you can see in the Lellinger's book, while he is trying to explain this particular enzyme, mechanism of enzyme activity. Now, if we look at, in 1971, the Protein Data Bank was founded with 7 structures, reaching 50 structures in 1979 and 100 structures 3 years later. But currently, this Protein Data Bank has got 29,564 structures. So you can easily understand the development from 1971 to now is such a huge amount of development. And this is actually what is possible because of the technological development. Now, if you look at the biochemistry research in the early 20th century, what fascinates Sir's lecture is how he introduced us about the glycolysis. And while introducing the glycolysis, always he tries to compare it with photosynthesis. So glycolysis is one hand, photosynthesis is on the other hand, and how these two are related to each other. In one case, we are looking at the oxygen, water as end molecule. In another case, it is actually the oxygen as end product. Now, while discussing about this, he came to understanding the development of the TCA cycle, the Krebs cycle. And here we could find that really the biochemistry, basic biochemistry understanding is actually strengthened one after another. On the other hand, if you look at the urea cycle, and from the urea cycle to the oxalate cycle, these are the very, why I have identified this, because these are the very few topics which are where Sir is used to give so much emphasis, and it fascinates us actually to understand. And these are the things why we get in love with biochemistry. And then, if you look at the viruses, so over here, you know that the first one is actually the tobacco mosaic virus, and its structure has also been determined. Then we can see slowly there are a huge number of structures and other things are coming up. We have the ureas, detailed studies of ureas, penicillin, the catalase, the myoglobin, and then these are the different things where we can find NADH, vitamin B12, FAD, ATP, thiamine, pyridoxine. All these are actually the components which helps us to understand these particular cycles in a very good way. And if we understand this, how actually it gets involved, the involvement of these are actually coming up much later, and how these are actually getting into each and every one, how the NADH actually is there in the Krebs cycle, how it actually works over here, and all the different things are is actually Sir used to explain to us with such a vigor, with such a passion, that it goes inside us. Now, if you look at from the secondary point of the mid and the late 20th century, that the protein secondary structure, then the chemiosmotic theory, and definitely after that it comes as ptosis, catalytic RNA, and definitely the central dogma, what is there. So when Sir is actually teaching us the central dogma, and it's only a couple of years back it's actually been established, but within this very short period of time, Sir used to explain it to us in such a way that I'm sure Arun you can remember Sir, how actually Sir act. He used to dance in front of us with respect to how in the ribosomes the protein actually gets translated. So then naturally the establishment of the double helical structure of the DNA, how it supported this particular central dogma, and these are the things which are coming. In the meantime, the protein folding, the solid phase peptide synthesis, and later on the innate immunity come into the picture. Now, when we actually gone into this DNA era, the DNA is there, and structure is there, central dogma is there, and this periodic table is, this particular genetic code is actually giving us something which reminds us of the periodic table itself, that from this genetic code, actually it is so interesting to find it out, so many different things. Just looking at this this genetic code, it always inspires someone that how we can actually look at our protein molecules, the structure of the protein molecules, the involvement of the different amino acids at the different sectors of the protein molecule. On the other hand, if you look at this, where these particular sequences are becoming very, very important, and Sanger sequencing developed, and how the Sanger sequencing helps to unravel that sequences on the DNA molecule, that is also coming up. And after the Sanger sequencing, the didoxy sequencing is also helped us to understand in a much bigger way of this particular DNA era. Now, here actually it comes that introduction of central dogma to the restriction endonuclease to give us this idea of molecular biology, understanding the molecular biology, the biology at the molecular level. And this restriction enzymes helps us to develop a concept regarding the recombinant DNA. And these developments in molecular biology revolutionized the enzymology and biocatalysis. For the first time, the DNA sequence of an enzyme of interest could be determined and cloned, and the enzyme could be overexpressed in E. coli for another suitable organism, and thus be obtained in sufficient quantities to be studied and used for industrial applications. The PSET is another thing which helps us after the didoxy sequencing application in a huge way. It comes this polymerase chain reaction. Then comes the directed evolution and the beginning of modern biocatalysis. Now, with the availability of recombinant DNA as well as the understanding of enzymes and their mechanisms, introduction of specific mutations into target enzyme was now possible. And the Nobel Prize in Chemistry 2018 was divided, one half awarded to Francis Arnold for the directed evolution of enzymes. Now, then comes actually, if we look at the how enzymes often exhibit promiscuous activity toward non-native substrates or reactions. By applying directed evolution, a specialist enzyme might be transformed into another specialist enzyme for the new activity at the cost of diminishing its original function. And you now know that this particular concept developed a completely new branch of biology, what we call as a synthetic biology, where the directed evolution is becoming very, very important. And in this overview, if we consider the whole thing, that here, you know, when we talk about the phenotype and the genotype, where we actually get something which comes out to our understanding how things are going to work out. Here I just wanted to mention that how the different new enzymes or enzyme modifications are actually done with the help of different types of methodologies like site-directed mutagenesis. Now here I'm just mentioning to you about something which is very important with respect to the early developments of this directed evolution. Now, if you look at that in 1967, Spidelman reported an in vitro Darwinian experiment using self-replicating RNA, which published in PNAS in 1967. And in the 1971, Eigen reports a theory of evolution of the molecular level, which actually been published in the Nature Wisson Skull Upton. And in 1980s, rational mutagenesis approaches of engineered enzymes show only limited success. In 1986, researchers at Synergy succeeded in the first directed evolution using an iterative rational mutagenesis approach. Noel's, the first true random mutagenesis by using the full sequence space. And 1993, the Arnold report the first iterative random mutagenesis. Here also random mutagenesis, here also random mutagenesis. But here the evolutionary concept is much more prominent to be explained. And then in 1997, Ridge and Jagger used directed evolution to improve the enantiomer selectivity of an enzymatic resolution. So how this particular directed evolution is being supported with our knowledge of the DNA and the mutagenesis, and then applying that is actually we could see from the development between 1960s to 2000. And over here, this is a little bit clumsy slides. I just don't want to go through all of them. Only few of them what I just wanted to mention over here. All of them are very important, but I just wanted to tell you that what you hear from Shankar Adhu's lecture regarding this 1994, here the therapeutic antibodies developed with phage display, and Shankar Adhu mentioned with respect to lambda, but this one is respect to the M13 bacteriophage. Then another one I just wanted to mention you about the organism-wide evolution through Mage 97 in 2009. This helped in a very, very strong way to give a background of evolution from the aspect of how we are going to look at it from the point of view of the change. And in 2016, the in vivo directed evolution and the artificial metalloenzymes are also something which are very important. But in the meantime, what we found that this CRISPR technology and the restriction enzymes are there, and now we have another technology, the CRISPR technology, which came out in 2020. It enables targeted in situ directed evolution, but in a much, much simpler way. And this is actually right now is one of the most hot topic to introduce, to do the recombinant DNA molecules. Now, so in one case, the mutagens, physical or chemical, are there. Then we are talking about the error-prone mutagenesis, and then we have the rolling circle amplification, or the RCA, and this RCA is actually the one which are also helping us in a very nice way of helping to develop this particular evolution of enzymes. I really wanted to target it with respect to the enzymes mainly because enzymes is something which is a very basic thing in the understanding of biochemistry. Now then the random insertional deletion strand exchange, the tandem repeat insertion, and these are the different methodologies which the development which helps us to do this type of approaches for the directed evolution of enzymes. And these, just look at it, just for the directed evolution of enzymes with the mutagenesis approach, how many different types of methodology developed. So this is what we call in biochemistry the innovative mind. If you have a creative mind, if you have an innovative mind, you can really contribute in a very big way in this particular subject area. Now, this is actually the future challenges and the developments what we can see with the customizable evolution of enzyme selectivity. The other one is the coupling of mutagenesis and selection, then evolution of inter-metabolic pathways, then obviously directed evolution of non-coding sequence. And what Professor Arun Datta already mentioned to you, that right now AI is also playing very important role in this, and this AI guided library generation is becoming very important right now for researchers. So what we can say that from the late 20th century to the early 21st century, what are the different areas which becomes very important is the ultra high resolution microscopy, the vesicle transport, the DNA repair. The first speaker who is actually one of the pioneers in working in this particular DNA repair process already presented how actually it is becoming very important. Then the high resolution cryo-electron microscopy, the telomere and telomerase, and the 3D structure of ribosomes. This is another area which actually gives an importance of the cryo-electron microscopy also. The iPS cells are becoming very important right now for the researchers, and the 3D cell biology is coming up in a very big way for doing a number of different types of experiments. The RNA interference as well as the structure of GPCR, these are something what we can say at the advances in biochemistry in the early 21st century. And naturally, in this particular time, what we can find out that the huge amount of sequence generation, sequencing been done, and Human Genome Project actually played a very important role with the help of that, there are next generation sequencing, and I mentioned already about the CRISPR-Cas, the integrative omics, the single cell mutations, and I mentioned to you about the synthetic biology now becoming very important and challenging for us. And this integrated multi-omics approach is actually with respect to transcriptomics, proteomics, metabolomics, genomics, multi-omics approaches are actually working in all the different areas, ionomics, phenomics, interactomics, and this is actually which is becoming very important right now, ionomics, with respect to these particular transports, and we could actually find out number of very important papers in this particular area. The other things what we can find it out, that it has got a huge impact on the health and disease. There are longitudinal data being actually developed, and then there is an integration. Here also I really thank Arun for introducing the machine learning process. The machine learning process actually gives an unsupervised integration of this, and we can really get this both unsupervised as well as a supervised multi-stage, multi-dimensional, which are actually helping us in the deep learning. The specific, I think one of the paper he mentioned in this particular area with respect to the help of this machine learning, deep learning to understand. And in the outcome, what we can find out that disease risk prediction, disease subtyping, biomarker discovery, molecular insights, and response to treatment, all these things are going to help us to understand the health and disease. So the rapid advances in the multi-omics enables deep phenotyping, which profiles the interaction of multiple levels of biology over time, empowers precision health approaches, and is poised to transform this future of healthcare. And that's why we, the biochemists, should actually concentrate us that how we can really do something for the society. Again, because of the time, I don't want to go through it here. I just wanted to mention about it, about the different discoveries. But what is actually very important over here, the Omics data, which are collected on the entire pool of molecules, represented as circles, except for the genome, all data layers reflect both genetic regulation and the environment, which may affect each individual molecule to a different extent. So the genome first approach implies that one starts from associated locals, while the phenotype first approach implies any other layer as a starting point. So here the genetics and biochemistry, genetics and molecular biology all comes together, shake hand with each other to actually become do the things which are going to be very, very successful. Again, I don't want to go through this. There are a lot of important informations available, particularly in the area of cancer. We could really look at that this big data from these approaches, the data analytics in life sciences are becoming very, very important. Now, big data is a term used to describe the large and the complex data sets that are difficult to manage and process using traditional data processing tools. Now, big data can be structured, unstructured, or semi-structured, and it can come from a variety of sources. Now, the technical advances in the 21st century, it facilitated this high throughput approaches and led to generation of huge amount of data and a dedicated approach to developing data analysis tools, which is becoming inevitable. So this is what we can say, that the system engineering, the modeling, the statistical tools, algorithms, machine learning, and artificial intelligence are now becoming integral part of biochemistry. So it is actually, there are a huge impact of these particular things in the biochemistry research. So there are a number of applications of these data analytics life sciences, particularly the model-based drug development, evidence-based medicine, public health surveillance, and the wearable device applications. And here, what we can find that there are large number of areas where this particular informations can go and really helps us to understand this. And this is where we can find the importance of the artificial intelligence algorithms plays very important role. And in protein structure prediction, already Arun Datta mentioned that it has got a huge one, and he mentioned about this AlphaFold, how successful they are. And we can see that this is actually for this artificial intelligence in protein structure prediction in the different models, in the AlphaFold 2 or ESM fold or the Rose TDF fold or the Open fold. These are becoming very, very important. And these are the things which are the areas which helps us to understand biochemistry from a completely different angle in the later one. So there are the AlphaFold protein structure database. Now, this is also the one where I just want to end my lecture, where Arun also given this lecture on this, the generative AI in life sciences. And in this particular case, this generative AI in life sciences is revolutionizing the various aspects of life sciences. It is accelerating the drug discovery, aiding in antibody development, enhancing single cells multi-omics models for disease understanding. The technology also plays a role in personalized medicine, which is very, very important right now, what we are talking about, the population genetics as well as the viral evolution, which is again something which is very, very challenging for us to know. With this, I just wanted to know that this, wanted to inform that this AI-driven technologies, particularly in this protein structure prediction, are significantly reshaping the landscape of drug discovery and development. By enhancing the precision and speed at which drug targets are identified and drug candidates can be designed and optimized, these technologies are streaming the entire drug development the processes. From whole systems to single molecules are actually coming up in a very, very big way in our field. And that is what the single molecule studies are becoming very important. This is another area where the biochemists are right now trying to understand a lot of different things. For the time, I just don't want to go for this. But this is something which is a very, very interesting thing. So with this, I just want to thank all of you for your hearing, and thank you.
Thank you, sir, for delivering such an inspiring and motivating lecture for the students. Now, Professor Dubhajuti Chattopadhyay will be felicitated by Professor N.C. Mondol with a memento and a potted plant on behalf of Professor Jagat Jiban Ghosh Foundation and Department of Biochemistry, Calcutta University. So, sir, a memento to Professor Dubhajuti Chattopadhyay, being presented by Professor N.C. Mondol, and a potted plant to Professor Chattopadhyay. Now, Professor Okay, now Professor Pranab Kumar Sarkar will be introduced by Professor N.C. Mondol for the inauguration of the exhibits, that is the memoirs of Professor Jagat Ghosh, kept at the Professor Jagat Ghosh Memorial Hall. Professor, I want to just mention a few words about Professor Pranab Sarkar, that Professor Pranab Kumar Sarkar gave the first Professor Jagat Ghosh Memorial Lecture in 2012 in this very department. He submitted his thesis under Professor Jagat Ghosh, although he did his PhD work under Professor B.C. Guha. But before submission of his thesis, Professor Guha was no more, so he did his final submission of thesis under the guidance of Professor J.J. Ghosh. And he also has submitted a write-up for the commemorative volume on Professor J.J. Ghosh, which is to be released on his birthday on 19 July 2025, and has been compiled by Professor N.C. Mondol. So with this, I request Professor N.C. Mondol to introduce Professor Sarkar.
প্রণবদার বায়োডাটাটা ইনসার বুক থেকে রিকভার করে একটা ফাইল তৈরি করেছিলাম, সে এখন দেখছি ওখানে নেই, আমার ব্যাগে নেই। এনিওয়ে আমি বাংলায় বলছি। প্রণবদা সেকেন্ড ব্যাচ, শঙ্কর মিত্রদের ব্যাচ। আর আরেকটা হলো হি গ্র্যাজুয়েটেড, দ্যাট ইজ বিএসসি অনার্স ফ্রম দ্য সেম কলেজ অ্যাজ আই ডু, দ্যাট ইজ হি ইজ my senior there. Anyway, after MS, he carried out research with Dr. Bishigho, but at the last moment, Dr. Bishigho died in 1962. Then he had to submit the thesis under J.J. Ghosh. This is one thing. Then he moved to California, J.T. Young. Therefore, he worked with Paul Doty, who was that time is the peak of his life. তারপর অনেক কাজ করে ফিরে এসেছিলেন 1966-এ, বোস ইনস্টিটিউটে 2 বছর ছিলেন। আমার এই পাশের কোল্ড রুমে উনি থাকতেন, কাজ করতেন, তখনই বেশি আলাপটা হয় আর কি, আমার তার আগে ছিল না। তারপর উনি চলে যান, তারপর ফিরে এসে আইআইএসসি জয়েন করেন যখন, তখন যোগাযোগটা হয়, ইনসা মিটিং, জেআরসি মিটিং সব কিছু। তাছাড়া সল্টলেকে থাকেন, ওনার বাড়ি মাঝে মাঝে যাই, উনিও এসেছেন আমার বাড়ি। তা হি ওয়াজ ইন দ্য অ্যাক্টিভিটি ফর এ লং টাইম। এখন কিছু প্রবলেম আছে, যাই হোক সেটা নয়, সে আসবে সবাই এতেই। প্রণবদা ইজ এ ভেরি অর্গানাইজিং ম্যান। এনিওয়ে, উইথ দিস আই ইনভাইট প্রণবদা to inaugurate the head hall. Yes. Hall. Okay.
Hello. I like to say a few words. The students, I think a number of students are sitting in Professor J.J. Ghosh Memorial Hall. If it is possible, please leave the hall, because our senior members, senior guests and the speakers will visit the museum now, which will be inaugurated by Dr. Pranav K. Sarkar. Now, I will request 20 to 25 persons can enter the museum at a time. Others may take their lunch packet, and the senior persons and the speakers and the delegates which will enter the museum now will get into the conference room to get their lunch there. Okay? Am I clear? So the students who are sitting there, please leave the hall. Sure. Oh, sure. Dr. Pranav K. Sarkar will say something about Professor J.J. Ghosh.
I don't want to take too long because I know all of you are hungry. Just 3-4 minutes, a few episodes about my interactions with Professor J.J. Ghosh. Okay? I first met him at my MSc class in 1957. And after 2 years of attending his classes, I really learned that he was an exceptionally gifted teacher, capable of getting the minds, capable of getting the attraction. Why? Because he has humor in his talk, he was eloquent, and he was really dramatic. As somebody else talked, I think Prof just talked about him, that he was doing drama as he was teaching from one side to another in the thing. Details will take a long time, but I'm not going to go through that. So 2 years of his classes make me understood about his quality as a teacher. Now at 59, I joined Professor Jagat Ghosh lab for doing my PhD work. Okay? After 3.5 years of working, Jagat Ghosh used to go to meetings very often. He went to Delhi to get instruments for biochemistry department. You know, he was the man who established the biochemistry department. And so he was very interested in the development of the department. So one time, after 3.5 years of my research, he went to Delhi and he was coming back. He saw a friend in Lucknow and he had a stroke. So he expired. Due to his premature death, you know, I had a serious problem. Who is going to supervise my thesis? I was ready to write my thesis, but there is nobody else to supervise it. At that time, our head of the department was Genji Niyogi. Why you say Genji Niyogi? Because Dr. Sudhir Niyogi used to wear Genjis in the department in the summer. So all the time, he was acting as the head of the department with wearing a Genji. So Genji Niyogi called me one day and said, Pranav, I have asked Jagat to supervise your thesis. You write up your thesis and Jagat will correct it. So that's how in 1963, I submitted my thesis under Professor Jagat Ghosh, and he was very nice enough to agree to write up the thesis. And then I was attending his classes with, joined the laboratory of, after 63, I went to postdoctoral work in USA to do University of California, and as Dhruva said, as I said, at Harvard University, spent a few years, and then came back to Bose Institute. But I went back for some reasons that is a very interesting story, but because of time, I'm not going to tell you. I went back to USA with a permanent visa and spent another 10 years working as a research associate with the University of Chicago and as a faculty at the Illinois Institute of Technology, Chicago. So, okay, when I came back, because Dr. Bachwat wanted me to develop the neurobiology department of ISCB. So I joined ISCB in 1978, okay? And my first student, Dr. Deeptin Chatterjee, published the first paper in 1983 at Journal of Neurochemistry. It's the isolation of astrocytes from rat brain, from glial cells. At that time, there was no procedure to do that. And it was one of the most important publication in my career, one of the most important publications. And Professor Jagat Ghosh looked at the paper and he came back from Science College to ISCB to personally congratulate me and Victor, and how much he loved science, you know. And he said, I will help you with more better students from now on. And he did really continue his zeal. And he was the only biochemical faculty who used to visit ISCB library every week, and to get himself updated with the recent developments, the latest developments in science and his area of biochemistry. And neurochemistry was one of the areas that he liked, that you know, he had done a lot of work with cannabis and all these things. So, in the end, I really feel fortunate to be associated with a teacher like him during the active part of my life. That's all I wanted to say about Professor Jiban. Let's go up to this. I hope I didn't take more than 5 minutes.
Students, you should carry the food coupons for getting the food, okay?
hello, check, hello, hello, check, check, hello, hello. আর একটু কমিয়ে দে, hello, 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 check, check, hello, 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 hello.
বাবুদা ওটো 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 বাবুদা ওটো বসে থাকে না, ওনার রেসপেক্টে উঠতে হয় না। হ্যাঁ আলো বন্ধ করে তো দেখতেই পাবে।
Hello, hello, hello.
Hello. Hello, here is a video projection. It was done by Ranojit De, actually Ranada, when sir visited Des Medical. And this is just 5 minutes projection. Then we will go to our right side, that is the bronze bust donated by Shankaradho. Then we'll go right again. There will be a box in which diary used by Professor Jagat Jiban Ghosh is kept there, and 136 pages of that diary has been photographed and mounted on the board. Then there is a cabinet where the applied chemistry division, when sir joined the university, then the applied chemistry division, biochemistry section, how it was look like. The photograph is there. The photograph is taken from the 100 years history of Calcutta University and the officers of Calcutta University during that period. Paddoja Naidu was the chancellor. Professor Siddhanto was vice chancellor, and others are given there.
inactive nature plant natural source activated লেগেছে তার সঙ্গে কোনো এদের বাড়াবাড়ি করতে দেয় activated কিন্তু free hand করে দেয় এই biological system it may একটা aggressive হয়ে যেত বোধহয় এই activated-টাকে আগে চেপে দেয় তুমি কাজ কর আর নো ফার্দার এই পর্যন্ত কাজ করবে এই বেশি কাজ করবে এই জিনিসটা আমার মনে হচ্ছে আমাদের এখানে বেসিক আইডিয়া দিয়ে কাজ করা activated co-activator দেখুন হরমোন টরমোনে আসছে অলরেডি রিসেপ্টর কো-রিসেপ্টর ওরা তো ওই দিক থেকে আসছে আমার মনে হয় আমাদের এই হরমোন কার্কুমিন আদার থিং এগুলো যেসব প্রিন্সিপালগুলো আছে সেটা আপনি যে একনলেজ সেটাকে আপনি এক্সপ্লেন করবেন জাম একটা কোয়ান্টাম জাম্পকে সিম্বাই ক্ষেত্রে হিউম্যান ব্রেন এত বলা যায় আপনি সিক্স জন্য ব্রেনে আকাশ পাতাল we still do not know what is the question and answer which is are there life in other universe? The Mars se deja primitive bacteria is involved. Mars se is ice. Bacteria extreme of bacteria. Extreme condition of সেগুলো কিনা একদম -25 -30 সেইগুলো মার্সে অভিযানে চলেছে আসবে তাহলে ডেভেলপমেন্ট আর কোথাও আছে কিনা মনে রাখবে যদি থাকে এখানেই ব্যাপার ডেভেলপমেন্ট অফ লাইফ যদি রুডিমেন্টারি লাইফ সব জায়গায় হেল্প ছিল ডেভেলপ করতে পারে কেন ডেভেলপ কিভাবে What a big question.
সবাই ছাত্রছাত্রী, ছাত্র নালে নামি থাকে।
সবাই এখানেই আগে চলে যান তাহলে সুবিধা
রইল আমাদের কাছে মানে একটা অ্যাসেট না পুরোটা তো পিডিএফ লাস্ট 82-তে না 80-তে জানা 80-তে জয়েন করেছি 84 থিসিস হ্যাঁ কাউন্ট করা যায় আমি তো মন্দিরের সঙ্গে এসেছিলাম তার সঙ্গে মনে নেই এসেছিলাম সেই আমার মনে আছে যোগাযোগ রাখি না আমার মানে আমার বাড়িতে সব বন্ধুদের আমি কোথাও কৃষ্ণ হানু কি কৃষ্ণ আপনি একদম মৈত্রী ম্যাডামকে দাও কাটতে অসুবিধা হচ্ছে এটা
We are going to start our next session soon. Please be seated. All the participants are requested to take their seats. Achha. So, Professor, PK Sarkar. Dr. P.K. Sarkar, who has inaugurated the memoirs of Professor J.J. Ghosh, just actor announcement, sir, who has inaugurated the memoirs of Professor J.J. Ghosh in the Professor J.J. Ghosh Memorial Hall. As he left us now, so his memento will be given to Dr. Shumantro Das by Dr. Shumantro Das will receive the memento on behalf of Dr. P.K. Sarkar from, okay, so yeah, yes. Dr. Shumantro Das will hand over the memento to Dr. P.K. Sarkar. Here is the memento for Dr. P.K. Sarkar, who has inaugurated the memoirs of Professor J.J. Ghosh. Now, Professor N.C. Mondol, chairman of the session, will introduce Professor Gauri Shankar Ghosh. But I want to just tell a few words about Professor Gauri Shankar Ghosh. Professor Gauri Shankar Ghosh is not just a lab-based scientist. He has done many outreach programs in India. He has encouraged college students for research. He interacted with students, as far as I remember, in Lady Brabourne College during his visit to Calcutta. He talked with them how they can go far with the subjects like biochemistry and microbiology. So with this note, I hand over the microphone to Professor N.C. Mondol to introduce our speaker, invited speaker, Professor Gauri Shankar Ghosh.
I am very much pleased to introduce Dr. Gauri Shankar Ghosh. He happens to be one of my students at MSc class in this department. He got his BSc degree in chemistry honors from St. Xavier's College in 1983 and MSc in biochemistry in 1985. Then he moved to Albert Einstein College of Medicine. He worked there for his PhD degree under Ladan Sulman. Then he moved to Yale University for postdoctoral work for 1 year. Then got appointment in San Diego. ও না না এইটা দিয়েছে ও এটা কাজ করছে না he then joined the Department of Chemistry and Biochemistry, University of California, San Diego, as assistant professor in 1995, where he was promoted to associate professor and professor respectively in 2001 and 2005. He is now a professor continuing there. His research activities have been dedicated to the investigating two major areas: understanding the mechanism of gene regulation by the NF-kappa B family of transcription factors, and second, exploring the role of serine arginine family of splicing factors in metazoan pre-mRNA splicing. Proceeding in this direction, Dr. Ghosh's group used multidisciplinary approaches involving X-ray crystallography, which is he has become a master of this thing, biochemistry, molecular genetics, and cryo-electron microscopy, the newly developed methodology for protein structure determination. In the first project, his principal focus was to understand every aspect of regulation, covering from I-kappa B kinase, which controls to the gene up to gene regulation by NF-kappa B dimers forming complexes with DNA target sites. Earlier, there was a lot of information available from molecular genetics and by cellular biochemical studies in this area, but there is a gap in vitro biochemical work to elucidate underlying mechanism at molecular level. Dr. Ghosh proceeded with an aim to understand the molecular mechanism involved in this process. They have established the 3D structures of NF-kappa B DNA and NF-kappa B IKB complexes, as well as the one domain of IKK1 alpha and IKK2 beta through X-ray crystallography studies. They have made a cryo-EM studies of IKK complexes to understand IKK2 beta activity switching from physiological to pathogenic states. They studied further to understand how these structural elements are involved in biochemical processes within the cell by in vitro experiment. They did in vitro experiments for this. They have studied, the above studies have contributed towards understanding of how degeneration of NF-kappa B activity, deregulation of NF-kappa B activity promotes cellular transformation. They extended the study further to understand the splicing regulation, focusing on SR proteins, SR protein kinase,
and spliceosome assembly, aiming at understanding as to how phosphorylation and dephosphorylation of SR proteins facilitate correct spliceosome assembly, emphasizing the importance of distinguishing features within the intron to prevent inappropriate assembly leading to abnormal cell growth. They extended the involvement of NF-kappa B and IKK in neurological disorders, specifically tauropathy and Alzheimer's disease. They have identified the key factors contributing to generations of Alzheimer's disease pathogenesis that include excess epinephrine, a deficiency in fructose 2,6 biphosphate, and heightened IKK activity. All these factors are affected. They studied to decipher the pathways associated with the aberrant activation of IKK2 beta that leads to a reduction in fructose bisphosphate levels, and unravel the disassembly of the IKK complex and the independent actions of IKK2 beta, particularly its localization within the nucleus of brain cells and its role in phosphorylating DNA repair enzyme, as well as the connection between elevated epinephrine levels and the dysregulation of IKK activity in the context of AD pathogenesis. They have developed a non-toxic IKK2 beta inhibitor capable of suppressing cytokine expression. This inhibitor holds promise for mitigating the pathological burden in various inflammatory diseases, including AD. They are on the way to determine cryo-EM structures of IKK complexes, aiming at understanding the transition in IKK2 beta activity functional from more physiological to pathogenic states. He will enlighten, I think, in today's lecture about this brain activity and AD disease. Dr. Ghosh has around 102 publications, 120 publications, and many book chapters and reviews. He has a long list of collaborators and PD associates. Honors and awards. In 1992, he got postdoctoral fellow of the Irvington Institute of Immunological Research. 1996, Young Investigators Award, University-wide AIDS Research. In 1999, Young Investigator Award, Cancer Research Program. In 1999, again, Alfred P. Sloan Fellow. In 2008, Best Scientist Award from the Irvington Foundation. 1999 to present, member ASAMB and ASCA. From 2004 and to 2010, member faculty of 1000. From 2008 to 2012, member NIH study section. Gauru Shankar যখন পাশ করে বেরোলো, ওদের একটা বড় ব্যাচ ছিল, নিরঞ্জন দাস, এরা খুবই ব্রিলিয়েন্ট গ্রুপ, অ্যাক্টিভ গ্রুপ। তো ওর সঙ্গে পড়ানোর সময় খুব একটা আলাপ হয়নি। পরে আমি যখন এনআইএইচে গেছিলাম ভিজিটিংয়ে শঙ্করদার ল্যাবরেটরিতে কাজ করতে, তখন আলবার আইনস্টাইনে এসেছিলাম একদিন একটা ল্যাবরেটরি ভিজিট করতে। সেই সময় ওদের ওখানে ছিলাম এবং পরিচয় হওয়া, ঘুরেছি অনেক। পরে তারপর তো আমি চলে আসি, ও আবার চলে যায় ওখানে। হ্যাঁ, হ্যাঁ, অমিতাভ ওদের ব্যাচ ছিল। অমিতাভ ও ফার্স্ট হয়েছিল। ও অমিতাভ পরে আমার ল্যাবরেটরিতে রিসার্চ অ্যাসোসিয়েট কাজ করেছিল কিছুদিন। যাই হোক, তারপর থেকে ওর সঙ্গে ও যখনই দেশে আসে আমার সঙ্গে দেখা করে, আমার বাড়ি আসে। ও এবং মালিনী দুজনেই, এবং আলোচনা হয় নানা রকম দেশের বিভিন্ন প্রবলেম নিয়ে ইত্যাদি ইত্যাদি। যাই হোক, তো আমি যখন একটা নন এনজিওতে কাজ করছি মেদিনীপুরের নামে, সেটা করছি শুনে হি বিকেম ভেরি মাচ হি ডোনেটেড 50,000 to this NGO. He is so wide-minded. Anyway, করোনার সময়ও আসেনি, কিন্তু যোগাযোগ হয়েছে, পরে এসেছে। এতে তোমার ওর গ্রামের বাড়িতে কিছু প্রজেক্ট করছে, সেগুলি দেখতে যাওয়ার কথা আমাকে বলেছে, একদিন যাব। ওর যে কোম্পানি করেছে, একটা বায়োটেকনোলজি কোম্পানি করেছে, চালাচ্ছে, কিন্তু খুব ভালো চালাচ্ছে, সেটা আমি দেখতে গেছিলাম, নিয়ে গেছিল। যাই হোক, এইসব ব্যাপারে হি ইজ ভেরি মাচ এনথুসিয়াস্টিক ফর দি extending his help towards the development of this society, especially the scientific society. With this, I invite Dr. Ghosh to deliver his lecture. Okay.
Okay, so let me start. And time is 2:37:49. My first goal is to finish in exactly in 30 minutes. Okay, I'm not going to take extra second. So I'll stop, even my slide remains. Okay. 8. Okay. And I have to thank myself first, because I'm the only speaker, probably self-invited myself. Okay. And all right, but I really sincerely thank the foundation, particularly Dr. Nag Chaudhary and all other members, right, keeping these memories of Professor Ghosh alive, and to really understand the young generation, the legacy of this department, particularly him and others. Really, this is tremendous. I haven't seen it, and I think this is very rare, probably even in Calcutta University. So he really had some truly devoted scholars, and so I thank you all. All right, and then I have to make a confession. So you all said that he was an amazing teacher, and you just heard Professor Dhruvajyoti Chattopadhyay, Dhruvoda, actually has his lecture notes still intact. And my confession is I never listened to his single lecture. True. So I had no idea what he was teaching. Why? Because I was just engrossed with his drama. So I come from a drama family. My father owned a drama company. And I used to think, man, this person could be an artist, right? And then he had an amazing character in a sense that outside the classroom, he really never looked around. So he never connected with us. Maybe part of the reason was his niece was in our class. Maybe that's the reason, maybe that's the character, maybe he was getting old, whatever it was. But in the class, in the dais, he was a different person. So I never really remember a single thing he said in the class, except he's really the drama. Single thing don't remember in terms of teaching subject. So with that, I felt really bad because every time we discuss about Professor Ghosh and every single student, my friends, he's an amazing person, and then I would just sit there and it's just like, why? Frankly, only in my second year, I really started listening to lectures till Professor Mundal started talking. It's a very difficult topic of genetics. So genetics is not intuitive to us. And so biochemistry was relatively easy. So you can go back and study the textbook and understand it. But genetics was really not like that. So it was difficult. So I had to listen to him. And he was slow. He was methodical. And so that changed a lot. Okay. But so here's my title, Mitochondria, Metabolism, and the Mind. All right? It's a very heavy title. I just came up with last night. And I prepared my presentation last night. I was busy till noon yesterday submitting 3 NIH proposals, okay, from here. So I came up with the title, and I thought this is appropriate, this seminar, to really, really remember him, because what I did not learn from his lecture, which I eventually have to learn now, because you cannot escape metabolism, and you cannot escape mitochondria. This is what I'm going to try to explain to you. All right. So first, and the most important slide is the acknowledgement slide. Here is like some people I'm going to really mention. So I'm just mentioning people who are involved in this project that I'm going to talk about. And I'm going to particularly talk about this person, Taposh Hajra. You heard this name from Dr. Shankar Mitra. He was my classmate. We used to sit, so I used to sit at the second bench. Taposh used to sit in the third bench. And Taposh has this personality. He's a good boy person. I was not a good boy person in the classroom, actually. I was a little bit uncontrolled, and Taposh was just absolutely what it makes a gentle person, right? And it was harder for me to understand how smart he was at the time, because we all used to live in the hostel, we were not going to studying, and I didn't understand it until he convinced me that you should work on DNA repair, right? And Taposh used to, I still call like 3 times a day. And then I was convinced. So, and I'm going to tell that story. And this is people in Taposh's lab, and Balaji Krishnan, Muralidhar, Rasti Gage, and Arvind Lidvan. These are the people that are involved in this, the neurological disease project that I'm going to talk about. And here's the people in my lab. And here's the person named Shanti Mundal. I'm going to tell you, you're going to hear his name a lot. And if not today, 10 years from now. This is a genius. Okay? And there's more about him, but not today. And these are other people also involved in this project. And these two guys, they're the master student, they're involved before. Okay. So let me tell you a little bit about the background for the students in particular. Really, I care about teaching students. I don't care too much about white-haired people like me, okay? But you have to really know these things. So Alzheimer's disease and related dementias is really very severe if you think about it. Worldwide, there's over 55 million people, they are really living with this disease. And so this is a short list. There are 600 neurological diseases, over 600. And I'm giving a list of only few. The Parkinson's disease, ALS, Huntington disease, ataxia, multiple sclerosis, stroke, epilepsy, and all of these things. A huge number of people suffer from various forms of neurological diseases. You can see this number in here. So what is really so we're talking about the brain, right? So these are the diseases of the brain, right? so it's basically the summary of the different parts of the brain. So all these diseases, they affect the different parts of the brain. That's why. So these are actually neurological diseases. The ultimate thing what happened, the neurons are impacted, right? And neurons will die by apoptosis. So these are the important facts, right? Brain is a very, very small part of our body, but perhaps the most important part of the body. We're talking about the mind, right? DJC talked about rational thinking, right? That's what differentiates us from others. So, for example, Alzheimer's disease affects, the disease initiates in the brainstem, and then will go to hippocampus, then will go to the whole cortex. And then you have Parkinson's disease, really, it will impact like cortex. And then you have Huntington disease impact like striatal neurons and so on. So they are really different parts of this, the body, right? So there are two major cell types. We're talking about the neuron and then glial. Three types of glial: microglia, astrocytes, and oligodendrites. So the large number of neurons, and that the connectivity of the neurons, that's what the important thing, right? That we get senses, and the senses is really passing through one neuron to another neuron and so on. And we don't understand it completely. Obviously, we understand very little. Glial cells are there. They're like 10 times more. They are to protect neurons. That's their primary job, okay? And so, so that's basically it about the cell type. And so I just talked about the neurotransmission. Probably you really study in your textbook or in the class that action potential, right? So you have, so neurons, so the textbook cannot do the justice of drawing it, right? So here is the cell body. Cell body is this in here, right? These are like dendrites, and this is axons. And here in the end, you have these synapses, right? So one neuron is connecting to this other neuron through the synaptic vesicle, through these synapses, right? And so the axon could be very, very long, could be sometimes short, could be very, very long, all right? So the information that's coming from somewhere, right, some cell or some neuron, through these axons, they had to travel really long. And the traveling happened through this action potential, meaning at the resting state, so we describe action potential in terms of voltage, millivoltage, right? In resting state, it's minus 70 millivolt because of this difference in positive and negative charge. So outside is, inside is negative, outside is more sodium positive. Sodium will come inside, potassium, more sodium will come inside, some potassium will go outside, so we are really changing the charge, right? And then until it hits closer to the synapses where you have really activated the calcium. So calcium will go inside, and then you have all these synaptic vesicles, and there the neurotransmitters are there, and they will really released to this in here in the synapses, and then you have the postsynaptic neurons should be activated by that, right? So that's the kind of basic biology that is known for a long time. And so I'm going to really talk primarily about the Alzheimer's disease, right? So dementia versus, so you really also really know. And many of us, or some of us, hopefully it will be none of us, but it will happen that some of us will suffer from dementia at some point in our life, at least some of us. And then, we always sometimes make a mistake that we all, when you get old and you forget things, is it dementia? No, it's not dementia. That's like normal aging. So if forgetting a name or, leaving sometimes your container in the oven, or finding right words, forgetting date or day, trouble balancing checkbook, losing keys or glasses, those are like normal aging process. A dementia meaning not recognizing a family member. That's dementia. Forgetting serve meal, that you prepared a meal that you forget that you have to serve it to people, right? Substituting inappropriate word, getting lost in own neighborhood, not recognizing number. putting iron in the freezer, and so on. This is a completely different thing, right? And these are very depressing. If you think your grandfather or your father or someone is suffering from that, that's devastating. But this number is really striking, that in 2000, right, as of 2024, right, that 9 million Indians are suffering from dementia, AD, ADRD, and kind of dementia. 7 million American, 55 million world population. But this 9 million, it was only in 2016, the number was 4. So from 4 to 9 happened in almost like 8 years, if the statistics is right. And the number is expected, unless there is some kind of prevention happening, in 2050, that number of suspected Indians would be 28 million. So 9 to 28. And Americans would be like 7 to 14. So it's a double for Americans, but for Indian, right, 3 times, right? So the disease is increasing in much faster pace in Indian population. You can say, because you are living, no, not the percentage. It is, oh, no, that's true. That's true, but still the ratio. But it's, unless like, population is increasing 3 times, that's not happening. So it's still is a higher number. Of course, proportionally, it's still less than the American. America has 400 people population, and India has 1,400 million people. But it is really troubling, though. The reason I say this thing, when I was growing up as a young person in a rural village, we never heard this word dementia. Of course, no one talked about this fancy English word, but it's not that people are not living old. I knew a lot of 80+ people. They are very sharp, right? But now I know many of my relatives, they're like 65 and 70. They are actually, some of them actually died of dementia, actually. So this is, so I see the difference. All right. Okay. I'm going to talk about the Alzheimer's disease. So the disease is marked by 2 things, that you will have primarily tangle formation, that inside protein aggregates, the primary protein called tau, or outside you have the plaque, is called A beta plaques, right? So those are the markers. It doesn't mean that every patient has to have these plaques and tangles. There's a disease, some dementia, you only have these tangles. Like tauopathy has a disease that you only have the tangles. So basically it's spread from, as I said, like from the brain stem to initially the brain stem, right in here, locus coeruleus, and then it's going to hippocampus, and then it's really going all over. If you really look at the kind of these brain images in here, you can see the holes. Means what? The neurons are dead, right? And they will never come back. Once it's gone, then you cannot regenerate the neurons. So why neurons are so important? Because neurons are terminally differentiated cell. Other cell, there are some terminally differentiated cell also, but most cells actually can regenerate, right? So neurons are that's extremely vulnerable. So that's why they have such a kind of elaborate protective mechanism of neurons, okay? So I'll just switch gear to DNA damage, right? So this is the subject of Sankota. This is the slide I got from Tapos. Okay, so there are 4 types of DNA damages, right? Primary number. You have what is called so you have the oxidative DNA damage, right? The base damage, and then you remove the base, and then you repair the damage, right? So here you have base excision repair, but base excision repair actually goes through a single-stranded cleavage. Then you have nucleotide excision repair. Here you have, let's say, thymine-thymine dimer that you have. Then you have double and single-stranded break. So this is DNA break. And then you have replication error. Then you have mismatch repair. So this is primarily the 4 damage and repair pathway. But I'm going to particularly focus on the double-stranded break because that's the most severe one. That once you have a chunk of DNA, right? You have a long DNA strand, suddenly a chunk in the middle is gone. How are you going to really fill it out unless you have a template, right? If it happened in the pair of, because you have pair of chromosome, if it happen in one, then you can use the other pair. But if it happen in both cases, or if it happen in long, so this could be the most detrimental one, right? And so one of these things happened in this double-stranded break is it lives with 3' phosphate. Okay? So if you have 3' phosphate, even if you fill it up from 5' phosphate to fill it up, because this 3' phosphate has to be removed, because it has to be hydroxyl so that DNA ligase can function, right? You remember all of those things? Okay, good. So there's an enzyme that carries out this function called polynucleotide kinase 3' phosphatase. So this is also really massively Shankar's work, Tapos's work, right? So they figure it out. So here you have, that, and I forgot to mention that this term ROS, I put in the green, either ionization radiation and ROS, that leads to this kind of strand break. And so polynucleotide kinase, PNKP, is the only enzyme that removes 3' phosphate. So that makes it very important. And so one of these things is that's what Tapos noticed. So Tapos started working on, he was also a basic DNA repair person. And he was focusing on double-stranded break repair. And then obviously we always happen, this is the beauty of doing science in America, which is you collaborate. Okay? I hope this is the story is the same in here too. But in America that you collaborate, and then people say like, you know what, I have this observation, I don't have this expertise, can you really do something? So Tapos, that's why got induced with, he has a colleague who was working on Huntington disease and ataxia. He says like, you know what, let's look at what happened to the DNA damage. And Tapos noticed that it really, in Huntington diseases, Huntington and ataxia, that the DNA double-stranded break sustains. So it's there, it's not repaired. And then he figured out the mechanism is PNKP is inactive. It's protein level the same. So it's not, the protein is still there, but it's not active. So that's what Tapos got me, hooked me up. He says like, what do you think what's happening? Okay, and that's what I got interested. So to long story short, that we figured out that there's an enzyme called PFKB3, right? So this is, again, so PNKP is a DNA phosphatase DNA kinase, okay? Now this PFKB3 is a carbohydrate phosphatase carbohydrate kinase. So what it does is, look at this glycolytic pathway. Now I'm coming back to Professor Ghosh, right? So I have to really redraw it, go to Google, go to textbook and say like, this is what's happening. What used to be very boring to me, suddenly the most favorite subject for me, not NF-kappa B anymore, because I feel I understand NF-kappa B much more. So here you have PFKB3. So this is the enzyme that synthesizes a small molecule, small metabolite called fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate, what it does, so this actually is an activator of this rate-limiting enzyme called PFK, phosphofructokinase. Phosphofructokinase converts fructose 6-phosphate to fructose 1,6-bisphosphate. Remember that? Okay, that's its function. But its discovery is also fascinating. It happened in 1983. I'm not going to talk about it, this enzyme, when people are trying to understand. Remember, the glycolysis was figured out in 20s, 30s, but it took another 50 years before they realized that something else was there. So what we saw, the association between PFKB3 and PNKP in the nucleus. So we're really looking at the nuclear genome, genome repair. So we're looking at the nucleus. So PFKB3 is this enzyme that's present in the nucleus, right? And so that was a hypothesis that definitely is fructose 2,6-bisphosphate has some relation to PNKP. And this is exactly what happened. And I'm going to, so this is an in vitro assay that Tapos developed. It's a synthetic, a single-stranded break substrate, where one of the strand here is this 3' phosphate and is radioactively labeled. So if you add PNKP, right, then you release phosphate. So this is a free phosphate in here. This is the substrate. What we found that if you take the AD sample, brain of AD sample, and then you say like, there is really no phosphate release. So nuclear extract of AD patient brain, like diseased brain, called post-mortem disease sample, is not there, okay? And so we knew that that's the problem. And we also really did measure that level of fructose 2,6 bisphosphate in the patient brain, and we found the patient brain has reduced level of fructose 2,6 bisphosphate. And then we also noticed that levels of PFKB3 in the patient brain is very low. This is the Western blot. You see, this is really disappearing, all right? So all of these kind of connections led us to believe that this is what's happening. Now, when you just supplement in vitro, so you have the patient extract, very little, right? Is it only 12%? And now you add more and more fructose 2,6 bisphosphate, you recover activity by 3 times more. That's all right? Something like that. So we also noticed that not only nuclear, now we are working on the mitochondria. Mitochondrial DNA is also damaged in AD sample. Remember, we didn't test only 4 samples. We tested 50 samples. I'm just showing you this example of only 4. So we tested between Tapos's lab and my lab, probably more than 50. So this is a huge number. So statistics is very, very strong. And you can really see it that mitochondrial DNA is, so this is a PCR assay. I'm not going to explain. This is a long PCR, LA PCR. The meaning that where the probability of the break is very high. And so you can see the long PCR mitochondrial DNA, and you can really see it that also some of these brain activated genes, their level is low. But this is really actively transcribing. And this is a non-transcribing gene, there is no damage. So there's a relationship between active transcription to double-stranded break and so on. So that, and we saw the exact same thing in SCA3, HD, ALS. ALS data is not there, so I'm not going to explain all of this thing, then I'm going to lose all the time. But you have to believe me, we see the exact same thing in 4 neurological diseases we have tested. All right, so that's the question. She asked the question, like, so thousands of people are working on ALS and neurological diseases. There's a lot of drugs people are really working on. There is not a single drug. Remember, whatever the drug we have, this is the cholinesterase inhibitor. That was people discovered in 80s, late 80s. Since then, there's no other drug for Alzheimer's disease. So, okay, so the question, so that's why NIH is really, NIH spent like last 20 years, $1 trillion on AD research only, hoping to have the drug. The many drugs people have developed, but they're all toxic. It's not working. All right. So, so we have this, basically it says all neurological diseases have the DNA damage. Is it DNA damage is really the cause of the neurological diseases or something there even before that? And so because it's a mitochondria, and we also, I'm not showing the data, we saw mitochondrial DNA damage happened before nuclear DNA damage, all right? So mitochondrial dysfunction is first before you have other thing. So then it is like, you know, and this is not only ours, there are other people also thought about it, ROS, reactive oxygen species. Why do you think of the reactive oxygen species to mitochondria? This is the reason, the electron transport chain, all right? So this is, you know this is her Jagat Babu's favorite subject. This is her, Dhrupada remembers beautifully recorded a 50 years old note, and I had to go back and read it again to really see that this is the problem was. Problem was, normal design was that you will have a proton gradient and you have the, see this, the electron transport. Whenever the electron is coming out, there is a proton gradient, proton will be in this inner space, and that proton will drive like ATP synthesis from ADP to ATP, right? So electron transport is very, very important. And then, you know, DJC also talked about NADH, NAD, and all of these important cofactors in biology that you need. You have the balanced amount of NADH, balanced amount of NAD. So these are different complexes. I'm not going to go through it. This is TCA cycle, all right? So this is what, so all the electrons that are really moving around, and this is what people believe, that this electron leakage. We don't know whether the electron leaks or is by design. Normally you have to have some ROS for the cell health. But this is, whenever you have this, really that superoxide, very transient. From there, you have hydrogen peroxide. And then, so, you have this enzyme to detoxifying it. You have the excess all of these things, you have this enzyme. So some way this enzyme get a little bit less active because of aging and all of these things. You slowly accumulate more and more of these reactive oxygen species, right? Primarily this, if they're accumulating, the most dangerous one is this hydroxyl radical. They last a little bit longer and they persist. So, so we designed an experiment. This is like, let's look at it, whether we can damage mitochondria and what's happening. And this is what you see. So this is actually cell survival versus rotenone is the complex 1 inhibitor. So you generate a lot more ROAs, right? So, and if you really do this ROS plot, initially actually ROS is good for the cellular viability. Initially, meaning the concentration is very low, right? When I talk about the initial, meaning you don't have that much. But then slowly that is really bad, okay? And if you really draw this curve like that, in a way that what happened is that this is a control, this is balance, the neutral zone. Above this neutral zone in here, ROS, is healthy. And when it's coming down in here, is unhealthy, okay? That is die. But if you divide within the healthy part, there is some part, the ascending part is really good. But as you're really growing more and more, you think you're still healthy, but damage has been happened. And you're really going down. So this is the curve. So then we also looked at this ROS. So here you have, this is basal ROS. This is just really staining DNA, all right? And then you generate ROS in 5 minutes. Green is the ROS, okay? And then you can see it, the green is slowly getting from here more to the nucleus. So it's really moved like a wave movement. We have a movie, but remember, I squeezed everything in 1 hour, right? Remember the ROS accumulation happened over, let's say 65. If people get dementia, that is happening over that many years. So it's really, really very slow process, which we have made it extremely faster. And so we see the disease case that more ROS, right, than non-disease. Then we did this. If we add fructose to 6-bisphosphate, then you actually can rescue this defect. Rescue this defect. So what you are really looking at this, a green is mitochondria. So in the disease cells, here you don't see any mitochondria. Mitochondria get destroyed almost, so not interacting with the mitochondria, right? In the left-hand panel, you can see there are like 4 cells, very little green there. And you add fructose to 6-bisphosphate, now you can really see a lot of green. Red, actually the tau aggregates, all right? And you can see is that tau aggregates is much lower when you add fructose to 6-bisphosphate. So fructose to 6-bisphosphate is doing something positive for sure, right? And we actually have this, the video you can really see the mitochondrial potential is increased. I don't know whether I can, you can, I don't know whether you can see that potential is increasing. That more and more get brightness. I don't know whether it's lost in conversion from my Mac to PC in here. But we also did it at another mouse brain with disease, AD, and you can see a lot. So the red is the aggregate. So there's a dye that recognizes tau aggregate, and you add fructose to 6-bisphosphate. And you really see much, much less that. So this is disease, and this is disease plus fructose 2,6-bisphosphate. And normally they go through the phosphorylation. I didn't tell you all the pathway that tau must be phosphorylated. Phosphorylation has been decreased dramatically. Here you also really look at it. The PFK3 level is going down. If you add fructose 2,6-bisphosphate, its level is going higher. So here you have in the disease sample that really much less green, there's more green, and so on. All right? And so this is the last experiment I'll show you. This is the data I got it this morning. they have the imaging data, they couldn't send it. This is the Western data. So what we really look at it, these glycolytic enzymes associates with mitochondria. So we purified mitochondria from brain, all right? And then looked at this glycolytic enzyme. We actually looked at 5 of them, but you can really see it. What we see that hexokinase 2, hexokinase is the first enzyme in the glycolytic pathway, and the level is much less. So this is like mitochondrial association. However, pyruvate dehydrogenase is not different. PKM antibody is bad. PKM1 that controls mitochondria has more than you have disease. So not all enzymes, some glycolytic enzyme associates with mitochondria, all right? And then we already showed that PKM also, not PKM, PFKV3 is also associated with mitochondrial membrane. So from there, the model is that here you have that a lot of this enzyme, glycolytic enzyme, they are associated with mitochondria. Whether they're, it's not that, you still have the cytoplasm had its own pool, right? So your main glycolysis is happening in the rest of the cytoplasm. But part of these enzymes are associated with mitochondria, and they have a distinct role, non-glycolytic role, okay? And what is the non-glycolytic role? We don't know. This is what we're trying to figure out, that it probably help in the ion transport. So they coordinate with this transporter, ion transporter, some way they coordinate it, so they probably kind of throw in fructose 2,6 bisphosphate, and not only fructose 2,6 bisphosphate, some other intermediate is also involved, we know that, all right? So this is the summary. I'm probably a few minutes more, right? 2 minutes, okay. So I'm not going to summarize it. So the question is, you already know, I already talked about it. So could this glycolytic intermediate be cofactor for the electron transport chain reaction? So this is like, these intermediates, their non-glycolytic function, they some way associate with this electron transport chain. So glycolytic enzymes should be there, and the cofactors generated by them is coming inside there regulated way. So what we believe is, so that's the function. So that's really the origin of it. Origin of the slowly you are accumulating it, right? So we always have to really look at the origin of the diseases. Otherwise, it's very, very hard to really tackle them. And of course, we are working on this molecule to develop it. If you think about, so my talk is done, but I'm going to really give you an analogy of it, right? When you really look at a system that is dysfunctional, right? That we have to look at the origin. So I'm going to give you sort of an analogy, like our society right now, right? He talked about it, the rational thinking. Do you really believe in India or in West Bengal, the people are rationally thinking these days, right? In a state where the government is killing its own people, right? Now you just think about it. Well, if you get rid of them, our society will change. No, you bring other parties, left front, BJP, Congress, they're going to really double down even more. Why? Because it's us. Right? This is like population. This is the population. The whole population has changed. Right? And so what's the problem? What is the problem with us? Right? Then you have to think about it. What's the problem with us? There's something problem. That's the first of all, you have to recognize there's some problem with us. So what is the problem? All right. I'm going to give you an answer. It's the food. What do you eat? It's the bread and meat. Okay? Bread and meat is the source of ROS. And we generate our environment and the weather, probably can take it. We are really accumulating ROS, we are behaving crazy. We are in a crazy state that we even don't mind killing our colleague, right? We are just afraid of everyone, right? I even don't know, I come out of this room and some of you think, who is this guy talking about it, right? I might be killed. All right. I think this is a little bit commentary, but in the disease context, if you think about it, you probably correlate with something. With that, I'm done.
Thank you, sir. Now, for this illuminating lecture, now Professor Gauri Ghosh will be felicitated by Professor N.C. Mondal with a memento and a potted plant on behalf of Professor Jagat Jiban Ghosh Foundation and Department of Biochemistry, Calcutta University. The potted plant to Professor Ghosh. So now, Professor N.C. Mondal, chairman of the entire technical session today, will be felicitated by Professor M.K. Poddar, President, Professor Jagat Jiban Ghosh Foundation. Professor Mondal will be felicitated by Professor M.K. Poddar today, and also with the potted plant. This is the memento, and also with the potted plant on behalf of Professor Jagat Jiban Ghosh Foundation and Department of Biochemistry, Calcutta University. Thank you, sir. Now there will be a reminiscence session which will be conducted by Dr. Aditi Nag Chaudhary, Secretary, Professor Jagat Jiban Ghosh Foundation. So I will hand over the mic to Dr. Aditi Nag Chaudhary for conducting this reminiscence session.
Come acting to record. You are the best on that.
There will be a reminiscence session to gather some of the views from the persons who were associated with Professor Jagat Jiban Ghosh. Okay, so this session will be very interesting to the students who will be able to know something about or some information about Professor Jagat Jiban Ghosh. So I'm handing over the mic to Dr. Nag Chaudhary.
Hello, good afternoon. We are already too tired, but we must discuss more. More, that is, those who are willing to remember Sir in a different way. I am calling Dr. Chandra Chaudhary, first Biochemistry Department initiator in undergraduate colleges, that is Ashutosh College. Chandra, please remember Sir in your own way.
Sure. Good afternoon, everybody. এখানে আমি জানি না আমার ছাত্ররা আছে কিনা আশুতোষ কলেজের কেউ ছাত্রছাত্রী থাকতে পারে হাত তুলেছে আমার এখন এই একটু আগেই আমার খুব মন খারাপ লাগছিল যখন ওই ঘরটাতে আমরা ওখানে আমাদের ফার্স্ট ইয়ারে ক্লাস হতো তো আমার একদম স্যারকে আমি চোখের সামনে দেখতে পাচ্ছি যেন উনি ওই পুরো এই গেট থেকে ওই গেট অবধি দৌড়ে পড়াতেন আমার ওটা একদম মানে চোখে জল এসে যাচ্ছিল আর কি ওটা মনে করে তো খুব সুন্দরভাবে অদিতিদি ওইগুলো করেছে মানে যেগুলো সুমন্ত্রদা মানে অসাধারণ কাজ হয়েছে ওটা যেটা স্যারের হাতে লেখা ওই দিকে দেয়ালে ওটা একটা দারুণ জিনিস হয়েছে আর আমার যেটা মনে পড়ছে আমি মানে স্যারের ফার্স্ট ক্লাসের কথাটা সেটা আমি যখন বায়োকেমিস্ট্রি পড়াতে যাই আমি প্রত্যেকবার ওই গল্পটা বলি সেটা হচ্ছে স্যার একটা গল্প বলতেন যে মানে যেহেতু বায়োকেমিস্ট্রির সঙ্গে কেমিস্ট্রির একটা কেন জানি না খুব অজানা লড়াই ছিল ওরা সবসময় মনে করতো দুটো নর্থ পোল সাউথ পোল মানে কেমিস্ট্রি খুব মানে করতো আর কি যে বায়োকেমিস্ট্রি কোনো সাবজেক্ট না যারা অঙ্ক পারে না তারা বায়োকেমিস্ট্রি করতে আসে এইসব আর কি তো সেই লড়াইটা আশুতোষে আমাকে প্রচুর ফেস করতে হয়েছিল তো আমি স্যারের গল্পটা দিয়ে প্রথমে শুরু করতাম স্যার বলেছিলেন যে প্রথমে বললেন যে তোমরা কেউ কেউ তুমি বায়োকেমিস্ট্রি পড়তে এসো, তোমার বন্ধুরা নিশ্চয়ই কেউ কেমিস্ট্রি পড়তে গেছে, তোমারই আরেক বন্ধু জুওলজি পড়তে গেছে কি বটানি, তাহলে তোমার কি মন খারাপ হচ্ছে যে তুমি এটা পড়ছো, বায়োকেমিস্ট্রি পড়তে এসছো? অত নাম করা সাবজেক্ট নয়, কেমিস্ট্রি একটা স্টলওয়ার্ড সাবজেক্ট। কিন্তু তখন স্যার বলতেন শোনো, এই গল্পটা তোমাদের চুপি চুপি বলছি, এটা হচ্ছে যারা কেমিস্ট্রি পড়তে গেছে তারা হচ্ছে ওয়ান আইড হরিণ, মানে সে একচক্ষু হরিণ। আবার যারা জুওলজি বটানি গেছে তারাও একচক্ষু হরিণ, কারণ যারা কেমিস্ট্রি পড়ছে তারা দে আর লুকিং ওনলি অ্যাট দ্য স্ট্রাকচারাল সাইড অফ দ্য অ্যাটমস অ্যান্ড মলিকিউলস, আর যারা করছে বায়োলজিক্যাল সায়েন্সে গেছে তারা শুধু ফাংশনাল দিকটা দেখছে। বাট উই বায়োকেমিস্টস আর টু আইড proper person, we are looking both into the structural functional aspects of life. তো এই গল্পটা দিয়ে আমি প্রত্যেকবারই শুরু করতাম, জানি না স্টুডেন্টদের মনে আছে কিনা, কিন্তু আমার মনে হয় এইটা প্লাস আজকে প্রফেসর ধ্রুবজ্যোতি চট্টোপাধ্যায় যেটা বললেন, ওই যে বুলারের এক্সপেরিমেন্ট, কীভাবে যে এই যে ব্রিজিং দ্য গ্যাপ বিটুইন কেমিস্ট্রি অ্যান্ড বায়োকেমিস্ট্রি, মানে বায়োলজি অ্যান্ড কেমিস্ট্রি, বা এইটা বা কেমিস্ট্রি ইজ দ্য লজিক বিহাইন্ড অল বায়োলজিক্যাল ফেনামেনা, এইটাকে মানে ওই বুলারের এক্সপেরিমেন্ট, এগুলো সব স্যারের পড়ানোটাই আমি আবার ওইগুলোই বলে দিতাম আর কি, কীরকম করে আস্তে 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 এই গ্যাপটা হয়ে গেল আর কি। সেইবারে উনি ওই ড্রামাটিক অ্যাক্টিং করে করেই দেখাতেন, যে ওইটাই বলেছিলেন উলার বোধহয় ওনার বন্ধু আরেকজন নাম করা সায়েন্টিস্ট, তাকে যখন উনি ইউরিয়াটা তৈরি করলেন তখন উনি বলছেন যে আমি ওকে লিখলেন চিঠি, সেটা সেই গল্পটা স্যার বলছেন যে এখন নাও আই ক্যান ক্রিয়েট ইউরিয়া উইদাউট দ্য হেল্প অফ কিডনি, মানে এইগুলো মানে আমি, আর তারপরে স্যারকে আমার মনে আছে আমি যখন আশুতোষে খুললাম, স্যার কিন্তু খুব ইন্টারেস্টেড ছিলেন, স্যার এসছিলেনও আমাদের ওখানে এনভায়রনমেন্টাল বায়োকেমিস্ট্রিতে, এনভায়রনমেন্টাল সায়েন্সের পোস্ট গ্র্যাজুয়েট ছিল, ওখানে ক্লাস নিতেন। আমি বললাম স্যার নেবেন? স্যার একদম বাচ্চা ছেলের মতো আনন্দে যে বলেন হ্যাঁ হ্যাঁ কেন ক্লাস নেব না, মানে আর আমাদের ওখানে প্রপার ব্যবস্থাও ছিল না যে স্যারের ক্লাস নেওয়ার মতো ভালো ঘর টর এইসব কিছু ছিল না, কিন্তু স্যার বলছেন তুমি একটা ঘর দাও আমাকে, আমি এক্ষুনি ক্লাস নেব, মানে এইটা মানে খুব মানে সত্যি কি বলবো, এরকম টিচার আর হয় না, মানে আমাদের বায়োকেমিস্ট্রিতে আসা স্যারকে দেখে, তাই না সুব্রত? কি এইটা খুব মনে পড়ছে। আর আমাকে বলতেন সবসময় ভিজুয়ালাইজ করাবে, হ্যাঁ টিচার অফ টিচার্স, মানে ভিজুয়ালাইজ করাবে, আগে শুধু অন্ধের মতো বই দেখে বলে গেলে হবে না, গল্প করতে হবে, গল্পটা বলতে হবে। ওই যে ফার্মেন্টেশনের কথা, আমার আজকে এইটা দেখতে দেখতে মনে হচ্ছিল যদি সেই গ্লাইকোলিসিসের ক্লাসটা কেউ রেকর্ড করে রাখতো কি ভাল লাগতো দেখতে, মানে গ্লাইকোলিসিস কি আমরা তো স্যারের হাত ধরে শিখলাম। হ্যাঁ ওই লুই পাস্চুরের গল্প যে ফরচুন ফেভার্স দ্য প্রিপেয়ার্ড মাইন্ড, ওইগুলো তো সব স্যারের কাছেই শেখা। আর কি বলবো, স্যারকে আমার অনেক শ্রদ্ধা জানাচ্ছি, মানে এরম মাস্টারমশাই জীবনে পাইনি আমরা কেউ। সবাই
থ্যাংক ইউ চন্দ্রা। Subroto Banerjee, batchmate of Chandra, I think you like to say something. Hey, energy. আরে লিখে এনেছো নাকি? লিখে এনেছো? তুমি কি লিখে এনেছো? ওরা হচ্ছে 81। হ্যালো চন্দ্র, তোমরা 81? 79। আরে ওরম দেখানো হবে নাকি? 100 জন লোক আছে, তার এখন দেখানো যাবে না, মুখে বলে, মুখে বলে।
মুখে বলবো না, মুখে আমি যা বলবো সেটা পরে শুনবে।
না, এখন এটা এত মুখে কি করে?
যে যা পড়ো।
তুমি বলো, সময় চলে যাচ্ছে।
আচ্ছা, ইউনিভার্সিটি ইন দ্য ওয়াল, ইউনিভার্সিটি উইদাউট দ্য ওয়াল। আই অ্যাম দ্য নাও স্টুডেন্ট অফ ইউনিভার্সিটি উইদাউট দ্য ওয়াল। আপনার যারা বসে আছেন সব ইন দ্য ওয়াল। যা ডেমো হলো, যা বলা হলো, সবই ইন দ্য ওয়াল বলা হলো। আমি কিন্তু ইন দ্য ওয়ালের মানুষ নই। আমি উইদাউট দ্য ওয়াল বলবো অ্যাবাউট দ্য জেজে ঘোষ। প্রফেসর জেজে ঘোষ আমার কাছে নেক্সট ফাদার। বায়োকেমিস্ট্রি পড়তে এসে ওনার কাছে প্রশ্ন রেখেছিল, আমি রাখিনি প্রশ্নটা, কেউ একজন রেখেছিল, স্যার বায়োকেমিস্ট্রি থেকে পড়ে কী হবে? চানাচুর বেচবে, তেলে ভাজা বেচবে, বাদাম ভাজাবে। এখানে পড়তে এসে আমাকে জিজ্ঞেস করতো তুমি কী হবে? সব বসে গেছি। আমি বলছি আমি চাটনি বা পাঁপড় ভাজা, এতক্ষণ যা শুনলেন না, বড় বড় খাবার খেয়েছেন আপনারা। আমি তো বলেছি আমি উইদাউট দ্য ওয়াল। তারপরে সব চুপ পড়ে গেলাম। উনি পড়াচ্ছেন, ওনার বাড়িতে আমি প্রায় যেতাম। খানাসি নাম্বার ওনার বাড়ি, আমার হাতিবাগানের বাড়ি। পড়াতে পড়াতে আরএনএ সিনথেসিস পড়াচ্ছেন। তো বলছে দেখো এই অ্যামিনো অ্যাসিড এই লেগে গেল, এই বলে একটা বলছে ডবল ডেকার বাস। তখন ডবল ডেকার বাস কলকাতাতে খুব চলতো। ডবল ডেকার বাসে আর জায়গা নেই, সে মানুষ ঝুলছে আর জায়গা নেই। পেছন দিকে একটা দৌড়তে মেরে এদিকে উঠে পড়লো, আরেকটা অ্যামিনো অ্যাসিড লেগে গেল, আর জায়গা নেই। ওমা দেখছি আরেকটা দৌড়তে পেছন দিয়েছে, মনে হলো ফাঁদের ছেলে ঠিক ঢুকে গেল। এইভাবে আরএনএ সিনথেসিস হচ্ছে। আমি ওনার বাড়িতে গিয়ে বললাম স্যার যেদিন কি আপনি থাকবেন না, কি মাইন্ড করবেন না, আমার ভালো ভাষা থাকে না, সেদিকে কিন্তু আপনার এই কথাগুলো আমার না রে। আজকে আমি 70 কাছে চলেছি, আমি আজকে এসে না এসে পারলাম না। যে গত 24 তারিখে মাথা ঘুরে পড়ে অ্যাক্সিডেন্ট হয়ে গেছিলো আমার, স্টিচ করি, কিন্তু আমাকে আমি না আমি গিয়ে এইখানে না হলে পারবো না। আপনারা দেখবেন এই কাজটা কী হয়েছিল। উনি যে ভার্সিটাইল মানুষ, উনি কী জানতেন না, এটা হোয়াট সায়েন্স ক্যান ডু ফর দ্য সিটি অফ ক্যালকাটা। আপনার বলুন, টিচার বলছেন যে সুব্রত এটাকে বলছে যে শুধু একাডেমি, শুধু মানে কি প্রসপেক্ট তৈরি করবে, শুধু ফিউচার তৈরি করবে, আরে কিছু কন্ট্রিবিউট করবে না। উনি এটা বানালেন। প্রফেসর রমেন পোদ্দার ভাইস চ্যান্সেলার ছিল সেই সময় আমাদের। আরবান মিনিস্টার ছিলেন ডক্টর প্রশান্ত সুর, আর অমিয় সেন ছিলেন আমাদের চিফ সেক্রেটারি। সবাইকে উনি ইনভাইট করলেন এই হলে বোধহয়। এখানে প্রশান্ত সুর বসে আছেন, অমিয় সেন বসে আছেন, আপনার আমার স্যার বসে আছেন, রমেন পোদ্দার বসে আছেন। তাদের মাসে আমাকে দিলেন বলতে ভোট অফ থ্যাঙ্কস। আমি চেয়েছি এইটা বানিয়েছি, এই যে চিঠিগুলো হচ্ছে, চিঠিগুলো উনি আমাকে দিয়ে লেখাচ্ছেন ওনার ঘরে বসে। গিরিদা মাঝখানে এসে বলে গেল স্যার ডাকছে, যাও ক্লাস শেষ হলে গেলাম, বলে এটা লিখো চিঠিটা। কী লিখেছো দাও। আমি বললাম কী হলো, প্রত্যেক লাইন কাটছেন। আমি বললাম অত কেটে কী করবেন, কালি ঢেলে দিয়েছেন ওর মধ্যে, আপনি কেন দিয়েছেন আমাকে? আরে আমি মা সরস্বতী মায়ের তলায় হাঁস হয়ে বসে আছি। দেখুন এটা দেখিয়ে আমার এটাকে, এখানে সুব্রত ব্যানার্জি নামটা অর্গানাইজিং সেক্রেটারি লিখে আছে বড়ো করে। এটা আমি করিনি, সিল স্ক্রিনে প্রিন্ট হয়েছে, সিল স্ক্রিনে প্রিন্ট। কেন তখন তো বামফ্রন্ট আমার পকটক করে টাকা বের করতো না, ও চিপুস পাঠিয়েছিল। ফলে টাকা দেবে কি আমাদেরকে এগুলো বানাতে? তো নিজের পকেট থেকে খরচা করে করছি। যে বছর আমাকে 300 টাকা দিয়েছিলেন সেই সময় হাতে। এইখানে দেখুন 15 জনের নাম আছে। 2 দিন ধরে ফাংশন হয়েছিল এখানে, এই খাওয়া দাওয়া হয়েছিল। আপনাদের লাঞ্চ ব্রেকে এখন রেজিস্ট্রেশন কত কী করেন, মোবাইল একটা টপ করে সব নিমন্ত্রণ করে ফেলছেন। আমাদের গিয়ে করতে হয়েছে। কোনো আমার বন্ধুরা বসে আছে, চন্দ্রা বসে আছে, আর 2জন করিন্দিরা বসে আছে, সব ফিউচার তৈরি করবে। আরে কী এখানে সময় নষ্ট করছিস? আমি কেন বলছি কথাগুলোকে? বলছি যে এই যে জেনারেশন তোমরা এখন আছো, তোমাদেরও কিছু সমাজকে দেওয়ার জন্য আছে। শুধু বায়োকেমিস্ট্রি পড়ে হবে তা কোনো, কত তো আমাদের টিচার হয়েছে, কত ব্যাংকে কাজ করছে, এরা তো বায়োকেমিস্ট্রি পাস করেছে। সবাই কি এখানে গিয়ে একবারে রাতারাতি হয়েছে? আমাদের রাজ্যে কজন হয়েছে? হয়তো 5 থেকে 6 জন, ম্যাক্সিমাম 7 জন, পরিচিত আমার 40 জন। তা সেই জন্যই বলছি, আপনাদেরকেও বলছি, তোমাদেরকে তুমি করেই বলছি ছোটোদেরকে, যে তোমরাও চিন্তা করো যে তোমাদেরও কিছু দেবার আছে, কিন্তু আমাদের তো এতজন প্রফেসর ছিলেন, তার মধ্যে কেন উনি বেছে নিলেন আমাকে? বলে এর মধ্যে একটা জিনিস আছে, ড্যাশিং জানে, পুশিং জানে। স্যার আমাকে বললেন যে সুব্রত আসো, এটা তোমায় করতে হবে। দেখুন আমার নামটা বোল্ডই ছাপা হয়েছে, এটা কিন্তু ইয়ের জন্য, আপনার সিল্ক স্ক্রিনের প্রিন্টের জন্য। উনি আমাকে বললেন আমার নামটা হালকা, তোমার নামটা মোটা কেন? আমরা আরে স্যার আপনাকে তো সবাই জানে, আপনাকে সবাই চেনে, মা দুর্গার মণ্ডপের সবাই মা দুর্গাকে দেখে, অসুরকে কি কেউ দেখার জন্য যায়? অসুর তো মা দুর্গা পায়ের তলায় থাকে, তো আমাকে একটু হাইলাইট করতে হবে না? তা আমি আমারটা মোটা করে দিয়েছি। ওনাকে কোনো পরিমাপে মাপা যায় না। আমি মা দুর্গাকে মজা করে বললাম, ওনাকে কোনো পরিমাপে মাপার মতন আমার কাছে নেই কিছু, কোনো ফিতে। আপনারা এটা পড়ে দেখতে বুঝতে পারতেন, কলকাতাকে হোয়াট সায়েন্স ক্যান ডু ফর দ্য সিটি অফ ক্যালকাটা। আর কারা এখানে এসেছে বুঝতে পারলেন, আমি আপনাদের বললাম এই সমস্ত দিকপাল মানুষগুলো শেষ সময়, আরে প্রথম সূচক 10:30টার সময় নিমন্ত্রণ করি, 10টার সময় এসে স্যারের ঘরে বসে আছে। স্যার নিচে নেমে দেখছি পুলিশ সিকিউরিটি টোটাল সব কিছু। স্যার উঠে দিদি চাটনির খাচ্ছে এরা, একটু মানা করো না, পাপড় ভাজা দিচ্ছি এবার। তো বলছে এই তুমি এতক্ষণ এসছো, সব এসে গেছে। আমি বললাম স্যার জানেন তো আমাকে, আমি লেট কামার। তো যাই হোক, আমাকে যখন বলতে বললো ভোট অফ থ্যাঙ্কস, হাতে করে কিছু পয়েন্ট লিখে নিয়ে এসেছি। কী করবো আমি অত আপনাদের মতন বলতে পারবো না। আর তখন আমি ফার্স্ট ইয়ার সেকেন্ড ইয়ার স্টুডেন্ট, আমার কি আপনার বাবা করে ইংরেজি আসছে, বাংলা মিডিয়ামে পড়েছি। আমি হাতে করে লিখে নিয়ে এসে বলছে ও কী হচ্ছে? আমি বললাম না দেখতে পারবো না তো, বলে এই বিদ্যাও আছে, আমার কিছু আছে। বাজার যাচ্ছেন, দেখুন উনি কোনো সময় আমি হাফ শার্ট দেখিনি, সবসময় ফুল শার্ট, আর কোনো সময় প্যান্ট জামা গুজে পরতেন না কোনো সময়। বাজার যাচ্ছেন ধুতি, বাড়িতে কোনো সময় অন্য কিছু পরতেন না, শুধু ধুতি আর পরতেও না ফুল শার্ট। হ্যাঁ, আর ফুল শার্ট হাতা লাগাতেন না বাড়িতে। তো আস্তে আস্তে বাজার গেছেন, হাতির বাগান বাজার, আমি ওখানে বাজার করি। তো ওনার পিছনে আমি দাঁড়িয়ে বাজারটা দেখছি, মাছের মাথা দুখানা নিল, মাছ তো নিলে মাছের মাথা। আমি বলি এটা কী ব্যাপার, একটা মাছ তো আমাদের দুখানা মাথা নিলেন কেন? আমি বাড়ি গেছি পেছন। ও স্যারের ব্যাগটা হাত থেকে কেড়ে নিয়েছি রাস্তার মধ্যে। আমি যাচ্ছি আর আপনি যাচ্ছেন ব্যাগ হাতে নিয়ে, তুমি কি রোজ আমাকে রোজই যাই? রোজ তো স্যার দেখতে না, আজকে তো দেখেছি ব্যাগটা দিন আমার হাতে। বাড়িতে গিয়ে বলছি স্যার দুখানা মাছের মাথা নিল কেন? বলে জানো না ও রোজ মাছের মাথা খায়। এই যে ব্রেন নিয়ে আপনার একটু কথা বললেন, আমি বুঝলাম যে ওনার মধ্যে কী আছে। আমি আজকে ওয়াটার ফিল্টার বানাই, উনানি আমাকে হাতে এইটা তুলে দিয়ে গেছিলেন। এসব তো এই যে ওয়াটার ফিল্টার এত তৈরি হচ্ছে, লোকে এত দাম দিয়ে কিনছে, কিছু করতে পারো না? আমি কী করবো? বলে দেখো না ওই ক্যান্ডেলটাকে কিছু করা যায় কি না? আমি কী করবো? বলে ওখানে সিলভার কপার ট্রিটমেন্ট করা যায়, সিলভারকে অ্যাক্টিভেটেড করবে, কপারকে অ্যাক্টিভেটেড করবে। ঠিক যে কী জিনিস? বলে স্যাকরারা সোনার রূপ গড়ায় না, ওদের কাছে গিয়ে দেবে সিলভার আর কপার, ওরা তোমার গালিয়ে দেবে নাইট্রিক অ্যাসিড, নিজে করতে যেও না, হাই টক্সিক নাইট্রিক অ্যাসিড। আর ওটাকে হিট দিয়ে নাইট্রিক অ্যাসিড টক করে ভেঙে যাবে, সিলভারটা অ্যাক্টিভেট হয়ে যাবে, কপারটা অ্যাক্টিভেট হয়ে যাবে। শোনো তোমাদের বলছি, এরকম মানুষ ছিলেন। আমি কিন্তু ওনার পায়ের মানে নিচেও একেবারে, কিন্তু কাকে দিয়ে যাচ্ছেন বীজ মন্ত্র চিন্তা করো। আজকে আমি সেটা করেছি, ওই সিলভার কপারকে হিট দিয়ে ভেঙে নাইট্রেট তাড়িয়ে অ্যাক্টিভেট করলাম। বুঝতে সিলভার কপার কী করবে? বলে RNA তো তৈরি করছে তোমার অ্যামিনো অ্যাসিড, অ্যাসিডের তো মধ্যে তো নেগেটিভ, আর সিলভার তো পজিটিভ, কপার তো পজিটিভ, নেগেটিভ পজিটিভের মধ্যে তো কলিশন হবে, সিলভার তো ওকে পেনিট্রেট করে ঢুকে যাবে, সেলটা তো মরে গেল। হরি বল। আপনাদেরকে বললাম শুধু বায়োকেমিস্ট্রির মধ্যে উনি থেমেছিলেন না, এটাকে ভাববে। আমার আজকে আসার কথা এই জন্যই, সেই 7 বছর দিয়ে আমি আসতে পেরেছি, উনি আমাকে নিয়ে এসেছেন। আগে প্রথমে চরণে মালা দিলাম, তারপরে দিদির সঙ্গে, আর দিদি আর এই স্যার পোদ্দার স্যার কী স্নেহ করে আমাকে ডেকে নিয়েছেন। আমরা স্যার আপনি কমিটির মধ্যে আমাকে ঢোকালেন কেন? সবই তো ডক্টরেট আর প্রফেসর, তা আমি তো কোনোটাই না, ও মানার মধ্যে তো আমাকে ঢোকালেন কেন? বাট তোমাকে দরকার আছে, কী দরকার বুঝেছেন উনিই জানেন, এনারাই জানেন। আচ্ছা নমস্কার, আজকে এখানে থামলাম। আর আমাকে বলার জন্য
এখানে সুব্রত, এখানে তোমার মতন আরও অনেক বিজনেসম্যান আছে ডক্টর অরুণ দত্ত বিজনেস করেন, ইন্ডাস্ট্রি করেছেন। এই ডক্টর গৌরীশঙ্কর ঘোষ ওরা বিজনেস করেন, ইন্ডাস্ট্রি খুলেছেন। হ্যাঁ, সবাই শুধু পড়াশোনা করছেন তা না। আমি এখন মৈত্রী দাশগুপ্তকে ডাকছি, কারণ তুমি স্যারকে শুধু স্যারের ক্লাস করতে, আর কোনো ক্লাস করতে না বলেছো আমাকে, চলে এসো।
আমার তো সবাই স্টুডেন্ট। আমি সত্যি ক্লাস করতাম না, আমি ডিসকলেজিয়েট হয়েছিলাম। আমি কোনো ক্লাস করতাম না, খালি টিটি খেলতাম। আর যেই স্যার যখন আমাদের এখানে টিটি বোর্ড ছিল, এবার শোনা যাচ্ছে তো, ওই টিটি বোর্ডের পাশ দিয়ে স্যার যেতেন, আমি হাতে ব্যাট বল নিয়ে স্যারের পেছন পেছন ক্লাসে ঢুকতাম। উনি আমার নাম দিয়েছিলেন পিং পং। এই পিং পং ক্লাসে ঢুকেছে। দিস ইজ মাই বায়োকেমিস্ট্রি ক্লাস করা। মানে উনি যেভাবে বললেন আমি তো ওটাভাবে বলতে পারবো না। আর বাকিদের ক্লাস করতাম না, সেটাও তো এখানে বলতে খারাপ লাগে না। আমি এনসিএমের ক্লাস করতাম। স্যার আমি আপনার ক্লাস করেছি না, খুব মন দিয়ে করতাম। না 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 স্যার জানেন আমি স্যার আপনি পরীক্ষা নিয়েছিলেন, আমি হায়েস্ট পেয়েছিলাম, আপনি বললে হবে না। আমি বললাম কিন্তু বাকি অন্য ক্লাস না করতে পারতাম না। কিন্তু আমি যে ঘোষের কথাই বলি, কেন করতাম, ওই প্রথমে তো ওই 2 আইড যে ব্যাপারটা, সেটা প্রথম দিন আমাদের ক্লিয়ার করে দিয়েছে না, একদম এক্স অ্যাক্সিস ওয়াই অ্যাক্সিস লিখে মাঝখান দিয়ে বায়োকেমিস্ট্রি, সো ইউ হ্যাভ বোথ দ্য আইজ, এটা ক্লিয়ার হয়ে গেল। এরপরে এই গ্লাইকোলাইসিস যেটা বলছে না, উনি যেটা করতেন, প্রত্যেকটা সায়েন্টিস্টকে বোর্ডে নিয়ে আসতে পারতেন। এইটা একটা অসম্ভব ক্ষমতা। মানে কাউকে যদি ওরকম লিখে দেওয়া হয় ABCDE, কারোর মনে থাকবে? কখনো মনে থাকবে না, ভালোও লাগে না। উনি একদম ডিরাইভ করতেন এভরি এনজাইম ডিসকভারি দ্যাট লেড টু দিস পাথওয়ে। উনি কী করে করতেন আমি জানি না, তাতে আবার TLC আঁকতেন। এই দেখো, হি ওয়েন্ট ব্যাক টু রুম, কুড নট স্লিপ. Then he came back next morning and he found the spot that showed that it was ribulose 1,5%. আমি এটা কেলভিন সাইকেলে চলে গেলাম গ্লাইকোলাইসিস থেকে, কিন্তু ওই লাইনটা আমি জীবনে ভুলতে পারবো না, যে একটা মানুষ এক্সপেরিমেন্ট ডিজাইন করে সেটাকে হাইপোথেসাইজ করে হোয়াট কুড বি দ্য প্রোডাক্ট, সেটা আবার TLC করে প্রমাণ করে সেই পাথওয়েগুলোকে জুড়ে দেওয়া। মানে একটা অদ্ভুত অভিজ্ঞতা, মানে যে কারণে ওই কোনো দিন অন্তত স্যারের ক্লাস আমার মুখস্থ করতে হয়নি। মানে স্যার এত সুন্দর করে পড়াতেন। এরপর এটা গেল হচ্ছে মেটাবলিজম পড়তামই না কোনো দিন যদি স্যার না পড়াতেন। এরপরে আসলো হচ্ছে মলিকুলার বায়োলজি। সেইটা আমরা যখন পড়াশোনা করেছি তখন তো মলিকুলার বায়োলজি জাস্ট মানে ইট কেম ডাউন, মানে 50s হচ্ছে তোমার ওয়াটসন ক্রিক সব হলো। আর তার আগের যে সময়টা, সেটা পুরোটা উনি বলতেন, পুরো ওয়েন প্রোটিন, মানে যখন মানুষ ভাবতো যে নিউক্লিক অ্যাসিড ইজ নট দ্য জেনেটিক মেটেরিয়াল, প্রোটিন ইজ জেনেটিক মেটেরিয়াল, হিস্টোনকে ভুল করে ভাবতো বিকজ হিস্টোন কনসেন্ট্রেশন ওয়াজ ভেরি হাই ইন নিউক্লিয়াস। এই গল্পগুলো যে কীভাবে বলতেন রূপকথার মতন, যে মানে বুঝিয়ে দিতেন যে হাউ সায়েন্স প্রোগ্রেসড আর হাউ DNA ওয়াজ এস্টাবলিশড অ্যাজ জেনেটিক মেটেরিয়াল। একটা জিনিস ভুলিনি, ওই অ্যাভারি ম্যাকাটি, আর বলো নামটা মনে আসে না, মানে এত সুন্দর, মানে প্রত্যেকটা জিনিস রূপকথার মতন বলতেন। কি যেন নিউমোককাস তাই না, ইঁদুরের মধ্যে ঢুকিয়ে, হ্যাঁ যাই হোক সব ভুলতে বসেছি, কিন্তু ওইগুলো এত সুন্দর বলতেন। আমি এখন স্যারের ক্লাসে বসে আছি। তারপর হলো DNA হয়ে গেল, তারপরে হচ্ছে আমার ট্রান্সলেশন পড়াচ্ছে। ট্রান্সলেশন যখন পড়াচ্ছেন, ওই যে উনি যেভাবে দেখাচ্ছেন, আমি তো নাটকটা করতে পারবো না যে এইভাবে RNA সিনথেসিস হচ্ছে। উনি ওইখানে বাইরে চলে গেলেন, গেটে বাইরে চলে গেলেন। তারপরে বললেন, গান করতে করতে ঢুকছেন, আমার হাত ধরে তুমি নিয়ে চলো সখা বলে, উনি অ্যামাইনো অ্যাসিডকে মনে পড়ছে, অ্যামাইনো অ্যাসিডকে টিয়ারেনের উপর বসিয়ে আমার হাত ধরে তুমি নিয়ে চলো সখা করে নিয়ে এসে বসিয়ে দেন। মানে কেউ ভুলবে কোনো দিন, মানে কি অসম্ভব। মানে আমি একটা একটা করে মনে পড়ছে বলছি আর কি। মানে আর আমার সব থেকে প্রিয় ছিল, যেটা একটু আগেই স্লাইডে দেখলাম, ওই কি যেন ইলেকট্রন ট্রান্সপোর্ট। মানে সত্যি বলছি, যখন আমি ভর্তি হয়েছিলাম তখন ওই বিএসসিতে থার্মোডাইনামিক্স, কাইনেটিক্স, মানে পিরিয়ডিক টেবিল ওয়াজ দ্য অনলি থিং দ্যাট ইউজ টু লাইক রিডিং। বাকি সবটা না হিব্রু মনে হতো। সবই পড়ছি, সবই লিখছি, ইকুয়েশন লিখছি, অঙ্ক করছি, সবই করছি, ভেতরে কিছু নেই। ফ্রি এনার্জি জিনিসটা দেখতে পাচ্ছি না। ওইভাবে পড়াশোনা করে যখন এসে বায়োকেমিস্ট্রিতে ভর্তি হয়েছিলাম না, কিন্তু অন্ধকার দেখছি যে কেন পড়ছি, সেইটা উনি দূর করে দিয়েছিলেন। সেই করে উনি যখন ওই ইলেকট্রন ট্রান্সপোর্টটা বলেছিলেন না, কীরকমভাবে বলেছিলেন, জলপ্রপাত, মানে ওপর থেকে পড়ছে, পড়বেই, সেটাকে হাউ ক্যান ইউ হার্নেস বাই ইউজিং ওই কী যেন বলে ওই, তাই না, তারপরে ওইটা করে কী করে তুমি হারভেস্ট করছো, অ্যান্ড ওয়াই ইট ওয়াজ নট ইন ডান ইন ওয়ান শট, মানে ওপর থেকে পুরো নিচে কোথাও ফেলতে পারতো তা নয়, কী করে হাফ হাফ ফেলে কেন ওইভাবে এনার্জিটাকে ট্র্যাপ করতে হয়েছিল, মানে অপূর্ব। তারপরে জাস্ট কমপ্লেক্সগুলো বসিয়ে দিলেন, হয়ে গেল। মানে কনসেপ্টটাই তো আসল, যে হাই মানে ওই মানে নেগেটিভ রিডক্স পোটেনশিয়াল থেকে, মানে সোজা কথা হাই পোটেনশিয়াল এনার্জি থেকে লো পোটেনশিয়াল এনার্জি আসবে, ব্যাস হয়ে গেল। মানে আর তো জীবনে পড়তে লাগেনি, মানে বাকি তো সবই যে ওই কাঠামোর মধ্যে ফেলে দিলেই হয়ে যায়। এই আর কি লিখেছিলাম বলো তো, মনে করার চেষ্টা করছি না, মানে আমি ক্লাসগুলো ধরে ধরে আর কিছু মনে পড়ছে না যাতে, ও না, তারপরে হ্যাঁ সে আমাদেরকে বললেন যে তোমরা বলো, তোমাদের ইউ হ্যাভ টু স্পিক আউট, টু বি আ সায়েন্টিস্ট ইউ হ্যাভ টু স্পিক আউট। আর আমি এসছি তখন, মানে আমি হোস্টেলে থাকতাম, গ্রামের না, মফস্বলের মেয়ে, মানে কোনো দিন দাঁড়িয়ে সামনে এরম বলতে হবে, মানে এখন যেমন অনর্গল বলে যাই, তখন তো সেটা একদম নয়। অনেক কষ্টে এসে স্যারকে বলেছিলাম, স্যার আমিও বলবো। এই যে এমকেপি স্যার বসে তো আমি খেয়াল করিনি, তো আমিও বলবো। স্যার এটা ইমাজিন করতে পারছে, আমি ওইরকমই মুখচোরা ছিলাম, এসে বললাম স্যার আমি বলবো। স্যার তো একটু মুচকি হাসলেন, কী বা বলবে। পরে আমার মনে আছে বাড়িতে গিয়ে সেই খাতায় লিখলাম, লিখে মশারির দড়িতে কাপড় মেলার ক্লিপ দিয়ে কাগজগুলো 3-4টে টানালাম। মা বলছে তুই কী করছিস? আমি না, কালকে আমার বলা আছে, আমি প্র্যাকটিস করছি। ওইভাবে লিখে লিখে প্র্যাকটিস করে এসে পরের দিন বললাম। কি একটা হাসি দিয়েছিলেন না আমি বলা শেষ করার পরে, যে হাসিটা হি ইজ টু কন্টিনিউয়াসলি গিভ মি। তারপর চিনে গেলেন আর কি আমাকে, না মানে করিডোরে আসছি যাচ্ছি সেটা, কিন্তু আলটিমেটলি হায়েস্ট পাইনি ওনার কথায়। সেই দুঃখটা আমার সারা জীবন থাকবে। অত ভালো পরীক্ষাও আমি কিছুতে দিইনি। 81-এ পাস করেছি। আই থিঙ্ক দিস ইজ, আমার স্যারের প্রতি, মানে আমি বলছি ওরকম টিচার আমি কোনোদিন দেখিনি। এরা যা বলছে না, আই ক্যান ইকো ইট আর কি। এবং যেরমভাবে আমি পড়াই, বেসিক্যালি এই যে এক্সপেরিমেন্ট করে আর দেখানো, যে মানে কি ডিরাইভ করা থিং, স্যারের কাছে এসে গেছি। ওটা স্যারই করতেন, আমি তো এইভাবে করেছি। থ্যাংক ইউ।
So here is the end of the Reminiscence session, and it was really interesting from all those who have shared their experience with us. Now, before the formal Thanksgiving, I want to inform you about the starting of this centenary celebration of Professor Jagat Jiban Ghosh. The centenary celebration started, as you have already heard from Professor M.K. Poddar, with the seminars at 3 different colleges having biochemistry honors under Calcutta University. The first program was held at Vidyasagar College on 31st August 2024. Professor Anirban Siddhanto, Department of Biochemistry, Calcutta University, was the invited speaker, and he delivered his lecture on intermediary metabolism. Dr. Subroto Majumdar, retired scientist, Bose Institute, was the chairperson of the lecture session. There was also student participation from 5 different colleges, namely Ashutosh College, Vidyasagar College, Vivekananda College, Gurudas College, and Shoshuna College in quiz and extempore. Extempore was judged by Dr. Subroto Majumdar, retired scientist, Bose Institute, Professor Anirban Siddhanto, Department of Biochemistry, Calcutta University, and Dr. Shopna Mukherjee, EC member, Professor Jagat Jiban Ghosh Foundation. The second program was held at Vivekananda College on 30th September 2024. Professor Rukhsana Chaudhuri, Chief Scientist, Retired Infectious Diseases and Immunology Division, CSIR IICB Kolkata, was the chairperson of the session, where the invited lecture was delivered by Professor Sanjay Ghosh, Department of Biochemistry, Calcutta University. A poster presentation competition was organized for the first, third, and fifth semester UG Biochemistry students. Evaluation of the posters were done by esteemed judges, and among the judges were Dr. Debnath Chaudhuri, retired professor and head All India Institute of Hygiene and Public Health, Kolkata, Professor Rukhsana Chaudhuri, CSIR IICB, Kolkata, and Professor Sanjay Ghosh, Department of Biochemistry, Calcutta University, for the first, third, and fifth semester, respectively. The topics of the posters were, for the first semester, antioxidants, nutraceuticals. For the second semester, probiotics, pre-probiotics, gut-brain axis, analysis of probiotics. And for the fifth semester, the topic was the different types of phosphatases in controlling biological function or system. So with this, we have the third program that took place at Ashutosh College on 30th November 2024. Professor Sanghamitra Sengupta, head of the Department of Biochemistry, Calcutta University, was the invited speaker. Her topic was the Gene Regulation Symphony, Harmonizing Genomic Instructions and Epigenetic Sculpting. The session was chaired by Dr. Sumantra Das, Treasurer, Professor Jagat Jiban Ghosh Foundation. There was a lecture competition on the topics in neurobiology for the first, third, and fifth semester students, respectively, from 5 different colleges. The lectures were evaluated by Professor Sanghamitra Sengupta, head of the Department of Biochemistry, Calcutta University, Dr. Ishani Deb, Senior Assistant Professor, Department of Biochemistry, Calcutta University, and Dr. Soumabroto Banerjee, Assistant Professor, Department of Human Physiology, Vidyasagar University. So special thanks to Professor Anirban Siddhanto, Professor Sanjay Ghosh, Professor Sanghamitra Sengupta, and Dr. Ishani Dev for being the part in the centenary celebration of Professor Jagat Jiban Ghosh. Also, we express thanks to the principal or TIC and the head of the departments of the 3 colleges who participated in this centenary celebration. Now, coming to the Thanksgiving part for today's seminar, I, on behalf of Professor Jagat Jiban Ghosh Foundation, want to say that we are extremely happy and excited to deliver our heartfelt thanks to Professor Shankar Mitra for giving his inaugural speech from USA in this seminar, and also to Professor Shankar Addo to deliver his online lecture. We are overwhelmed to see how you paid your tribute to Professor Jagat Jiban Ghosh. We are grateful to Professor Arun Datta, Professor Dhrubajyoti Chattopadhyay, and Professor Gaurishankar Ghosh for delivering lectures in this auspicious occasion. Our thanks are due to Professor Pranab Kumar Sarkar, Senior Scientist, CSIR IICB, for inaugurating the memoirs of Professor Jagat Jiban Ghosh, kept at Professor Jagat Jiban Ghosh Memorial Hall, where all the exhibits dedicated to Professor Jagat Jiban Ghosh were collected by Dr. Aditi Nag Chaudhuri, Dr. Debnath Chaudhuri, Dr. Sumantra Das, and Dr. Amit Krishna Dev. We are also thankful for the video of Professor Jagat Jiban Ghosh provided by Dr. Ranujit Dey of Days Medical. We express our extreme gratitude to Professor N.C. Mondol for chairing the whole technical session in the seminar, and also to the participants who have taken part in Reminiscence, like Professor Maitri Dasgupta, Subrata sir, and Dr. Chandra Chaudhuri. So thanks to all of you for sharing your views regarding Professor Jagat Jiban Ghosh. We are highly thankful to all the delegates and participants, especially the students, research scholars, for attending this seminar and making this event successful. On behalf of Foundation, I want to express thanks to the head of the department, Dr. Sanghamitra Sengupta, Department of Biochemistry, Calcutta University, for collaborating with Professor Jagat Jiban Ghosh Foundation in organizing this international seminar. Thanks are due to all the faculty members of the Department of Biochemistry, Professor Moitri Dasgupta, Professor Sanjay Ghosh, Professor Anirban Siddhanto, Professor Prashanto Kumar Bag, Dr. Onindita Ukil, Dr. Gitanjali Sundaram, Dr. Ishani Dev, Dr. Alok Ghosh for extending their wholehearted cooperation and support for taking care of the different logistics required for this international seminar. We are thankful to all the students and research scholars of the Department of Biochemistry for their role as volunteers in different committees. We also extend thanks to all the non-teaching staffs who have taken utmost care to help to make this program a grand success. Efforts of all the members of Professor J.J. Ghosh Foundation for arrangement of international seminar and decoration of Professor J.J. Ghosh Memorial Hall with his memoirs to celebrate his birth centenary are worth mentioning. Thank you all. And one announcement will be made by Dr. Aditi Nag Chaudhary regarding the certificates. And after that, there will be tea and snacks.
All the certificates will reach you through your email ID, which are also kept with us. You will get it within 7 days. Okay? Thank you.
So this is the end of the session of today's seminar, and outside tea and snacks are awaiting. So please have it. Oh, okay. Achha. Okay, this is the end of the session. You will be getting your certificates online. There will be a photo session now with the organizing committee.